Do we know how to share our screen? Is it obvious from this platform?
Yes, you can share your screen. So in your right side, there is this present now option. If you're using a laptop, then you will be prompted with the tabs that you want to share. So go to your right, and then you can see this present now window. So if you click present now, and then you'll be prompted with the options to share from a list of tabs. So if you want to share your entire screen, then you can share your entire screen. But if you don't want to share your entire screen, you want to share a tab, then you can do that as well.
Okay, I've got you're presenting to everyone, stop presenting. Okay, right. So I'm presenting, but what you can't see my screen at the moment.
I can see your screen, I guess. So if you open your PowerPoint presentation, then we'll be able to see that.
Okay, so that's my PowerPoint presentation. Can you see that?
There is some time lapse, I guess.
Probably, yeah.
Your Google Meet tab is open. We can see your Google Meet tab and your computer as well. It is 1349. So I think you'll just have to click on that Microsoft PowerPoint icon in your right. So it's Google Chrome and then Outlook and then Microsoft PowerPoint. So you'll have to click that one.
Can I just say one thing? I'm still saying I'm presenting. Is that blocking you, Nick?
That could do that.
I'll stop presenting on mine.
Okay.
Okay, that might have been an issue.
Right. Okay.
Okay,
right.
now you can do it right now, I think.
Right. Just talk through what I do.
You go to present now.
So I don't have present now anymore on the screen. How do I get that?
Can you see present now, or you are presenting?
I've got it now.
Okay. I've got it.
Now your entire screen. Okay. So do I want to share my entire screen, a window, or a Chrome tab?
I think you should go for your entire screen.
Okay, that's what I'm trying to do, but it won't. So the share button is grayed out, so it doesn't want to let me share the screen.
I cannot say what's happening with you. I think you should try stop presenting. Is there an option to stop presenting?
I have no option to stop presenting, only to start presenting.
And then you start presenting.
Okay. Still not letting me share my screen.
So you'll have to select your entire screen. From the drop-down menu, there is, I think, your Chrome tab and stuff. And then you'll have to select that one. So if you do not select that one, then you'll not be able to share that screen.
I've selected it, and then it says share your entire screen, but then it won't let me click that button. What we might have to try is me dialing in from another computer. Maybe I might try just using a different computer, because this doesn't seem very happy.
You could do that, but you'll have to do it very quickly.
It will take me 2 minutes to go and swap computers.
Then you do that. This is much better. Or you can leave the meeting and then you can re-enter the meeting to check if that works here. That way you can do that as well.
Okay.
Assalamu alaikum, sir.
Yes, please.
Sir, আমাদের এখান থেকে 7টায় অটোমেটিক্যালি ফেসবুকে স্ট্রিম অন হয়ে যাবে। এখন যে উইন্ডোটা আছে এই উইন্ডো নিয়ে অন হয়ে যাবে ফেসবুক স্ট্রিমিং, সেটা অটোমেটিক। আর ইউটিউব স্ট্রিমটা আমরা চাইলে এখন করতে পারি, এখন অন করে দিতে পারি, অথবা কিছুক্ষণ পর করতে পারি, দুটোই চলছে।
পরেই করে দাও তাহলে। আর আমাদের যেহেতু নিক এবং জর্জ দুজনই চলে আসছে, তো 7টায় তুমি শুরু করে দিও।
ওকে স্যার।
নিক কিন্তু এখন কানেক্টেড নেই।
নিককে লিভ নিতে বলেছি যাতে আবার জয়েন করতে পারে।
ফেসবুক লাইভ যেহেতু অন হয়ে যাবে, আমার মনে হয় তুমি তাহলে নিক আসা পর্যন্ত বাংলায় কিছু কথা বলতে পারো, তারপরে নিক আসলে তো অফিসিয়ালি তুমি স্টার্ট করে দিও।
Okay, so we are live right now. Good afternoon, good morning, good evening, everyone. We will start after a while because our wonderful speaker, Dr. Nick, is not here with us. Judge Lomonossoff and others are joined with us in the panel. So please wait for a while because Dr. Nick has a very tight schedule. Just bear with us. So we are live on both the platforms. We are live on YouTube and we are live on Facebook. And we are waiting for Dr. Nick to join us. So Eid Mubarak to all of you. If you don't know what Eid is, but this is like Christmas season for Muslims and we're celebrating it. And that's why we received a little less registrations than we are used to. So we're used to 700, 800 registrations per event. But this time it was a bit less because, we could do promotions for 2 days. And that's it. We went for the promotion on August 3rd, and it's just 2 days, and we could reach out to as many as 23,000 people. That was quite a huge number of people that we could reach out to, but We have received around 400 registrations for this event. So we expected 1,000 registrations. So if we could do promotions for say a week, then we could get as many as 1,000 registrations. But we're running short on time. So a lot of people have joined us at the panel, but we are waiting for the Nick to join.
Can you hear me?
Yes, we can hear you. Yep.
Okay. Can you hear me and see me?
No, we cannot see you because your camera is turned off, I guess.
Okay, let me just
Okay, it's on.
let me just sort that out.
Yeah, we can see
Okay. Okay, so let's try and see if we can share the screen on this computer. Entire screen. Allow.
Yep, we can see your screen.
Yes.
That's perfect.
You can see the screen. Okay,
Yeah.
right. Now I just need to get the presentation up.
You'll just have to open the Microsoft PowerPoint presentation, and that's all.
Yeah, but the presentation's just moving from one computer to another. Yeah, it's just going to take a few moments because in order to load the file, I think, because it's a very large file. But it looks as though this is going to work.
So that's perfect.
Okay.
So we can start right now.
Yes, please.
Yeah,
What do you say?
So I have one question. So when I'm
Sure.
presenting, do
Yeah.
you still see me?
Yes,
Do
we can
still
see you.
me?
So there is an option to turn your video camera off, so you can turn your video off if you want to.
Yeah, I'll keep it on.
Okay, that's perfect. Okay, let's start. Bismillahirrahmanirrahim. In the name of Allah, the most gracious, the most merciful, I would like to start Shaping the Future virtual seminar on vaccine development, entitled Virus-like Particle Vaccines Made in Yeast Against Polio and Emerging Diseases, which will be covered by Dr. Nicola J. Stonehouse, and the Transient Expression for Vaccine Production in plants, which will be covered by Dr. George Lomonossoff. So, Assalamu alaikum, Eid Mubarak. If you don't know what Eid is like Christmas for Muslims, a week-long celebration. So, good morning, good afternoon, good evening, everyone, depending on wherever you are watching us live on Google Meet, Facebook, and YouTube. I'm your host, Sam Shurka from Bangladesh. Welcome to the seminar. There are 3 primary ways you can participate in today's event. The first 80 registered people have joined us at the panel by Google Meet. Others are watching us live on Facebook and YouTube. So if you face any difficulties in one of the platforms, then please consider trying another. For Facebook, please visit
short.ai.world/spb02.
Or you can watch us live from SciencePortability's official Facebook page at fb.com/SciencePortability. Now for YouTube, please visit short.ai.world/spb03. You can find all the links at SciencePortability's official Facebook page at fb.com/SciencePortability. Dear Google Meet attendees, please make sure to keep your video and microphone turned off other than the panel. And for panel, please mute your microphones other than the speakers so we do not get any background noises and participants have no difficulty understanding us. Dear attendees, please put on your headphones for the best ever experience. But still, if you face any difficulties whatsoever, please let us know in the comment sections. We'll try to fix them as soon as possible. First of all, I'd like to thank our partners and sponsors. Shaping the Future virtual seminar on vaccine development is brought to you by SciencePortability, sponsored by Cell Genetics and Plant Biotechnology Lab. SciencePortability is a scientific communication platform based in Bangladesh. Members are highly enthusiastic to explore and do science. We communicate and collaborate with other interested groups and people who are striving for a common goal from all around the world. This has been our 4th international initiative. Our first Shaping the Future virtual seminar on genome editing entitled The Basics of Genome Editing, What, When, How by Dr. Kutubuddin Alimullah was a huge success. We had 716 registered participants from more than 20 countries, followed by a second successful one on the production of 4th generation biofuels entitled Microalgal Cell Factories, the 4th generation perspective for industrial biofuels by Dr. Pavan Dr, where we had 453 registered participants from more than 4 countries, followed by a successful third one on speed breeding technology entitled Speed Breeding Crops to Feed 10 Billion by Dr. Lihiki, which we covered just the last week, where we had a whopping 611 registered participants from all around the world. All of them were highly praised by the scientific community as well. You can watch the recorded video of our first 2 events on our YouTube channel. The third one is in post-production. Will be available on YouTube and Facebook within a week, so stay tuned for that. If you want us to host more seminars like this one, then please make sure to like and follow our official Facebook page at fb.com/SciencePortability. Subscribe to our YouTube channel at short.ai.world/spb_YouTube and hit the notification bell icon to be the first to get the latest updates on our upcoming seminars. I would like to thank all the organizations that have cooperated fully throughout our promotion. I would like to thank our university partners, content writers, translators, social media team, streamers, and print media partners. And finally, I'd like to thank you, the audience. We were overwhelmed by your responses. We have received 383 registrations from all around the world. Without your continuous support, we couldn't be here today. It would not be possible without the active contribution of Science Polio Bangladesh team. So a big round of applause for you guys as well. So without further ado, let's introduce you to our panel. Let's meet today's event chair, Dr. Abdullah Muhammad Shual, Professor of Biotechnology and Genetic Engineering at Jahangirnagar University, Bangladesh. Dr. Shual is a respected authority in the field of biotechnology. But for those of you who don't know him, he's the director of Cell Genetics and Plant Biotechnology Lab that is well known for its high research output. He did his PhD in agricultural biotechnology from Twinbok National University, South Korea. He had worked as a research collaborator for the International Rice Research Institute in the Philippines. He was awarded the prestigious JASPS postdoctoral fellowship. He had worked as a postdoctoral research associate at the Cell Genetics and Citrus Improvement Lab at the University of Florida. Dr. Shual has published articles in many reputed international journals. We are honored to have two distinguished researchers from Bangladesh as our esteemed panelists. Dr. Mohammad Morsal Billah, a former visiting researcher at the University of Edinburgh, now a professor of biotechnology and genetic engineering at Kaplan-Meier University in Bangladesh, and Dr. Mohammad Mainul Hassan, a former postdoctoral research fellow at the University of Birmingham in the UK, now the head of research and development at Inactivated Vaccines Limited, will join us in the discussion. Along with a prominent researcher in plant biotechnology and our event chair, Dr. Abdullah Muhammad Shual. And now, the attraction for today's event. We are very delighted to have not one, but two of the world's leading experts in vaccine development. One is Professor Dr. Nicola J. Stonehouse, a fellow of both the Royal Society of Virology and the Royal Society of Arts, a professor of Molecular Virology at the University of Leeds in the UK. The other is Professor Josh Lomonossoff, project leader of biological chemistry at John Innes Center in the UK, have joined us from England at our mega event, Shaping the Future virtual seminar on vaccine development. Welcome, both of you. Both of them are well-known and respected figures in the field of virology, but for some of you who don't know them, Professor Dr. Nicola J. Stonehouse is a fellow of both the Royal Society of Biology and the Royal Society of Arts. She is interested in virology and RNA biology, applying novel approaches to the study of virus replication and assembly in two main research areas. Coronaviral replication, especially the role of the viral polymerase of foot and mouth disease virus, founded by the Biotechnology and Biological Science Research Council, that is BBSRC, using structure and evolution-based methods in the design of novel vaccines. For example, Polio and Antivirus 21. The work is funded by WHO, Gates Foundation, and the UK Medical Research Council, that is MRC. Dr. Stonehouse teaches at undergraduate and postgraduate level, contributing to a total of 15 modules, and acted as the chair of examination group of the University Graduate Board until 2016. She currently supervises 5 PhD students, co-supervises a further 2. In addition to acting as an internal examiner over 10 times, Dr. Stonehouse has undertaken PhD examinations in the UK at the University of Cambridge, York, and Glasgow. She has also examined PhD theses from Spain, Malaysia, and Australia. Dr. Stonehouse is a fellow of both the Royal Society of Biology and the Higher Education Academy, and was elected to join the Council of Microbiology Society in 2016. She has acted as a member of grant review panels for MRC, Wellcome, the Royal Society, and BBSRC. To know more about Dr. Nicola J. Stonehouse, you can Google. She is a prolific figure in virology. You'll get her Wikipedia page, that is wikipedia.org/wiki/Nicola_Stonehouse. And Professor Dr. Josh Lomonossoff graduated from the University of Cambridge in 1976 and studied for his PhD at MRC Laboratory of Molecular Virology, that is LMB, Cambridge. He subsequently moved to the Johns Innes Center, Norwich in 1980 and has continued to work there ever since, apart from 2 periods of sabbatical leave in the USA, Cornell University and the Scripps Research Institute. George's research has focused on the molecular biology of RNA plant viruses and their use in bio and nanotechnology. He is an honorary professor at the University of East Anglia and Nottingham and has coordinated several EU framework consortia. In 2012, he was named BBSRC Innovator of the Year for his work on plant-made pharmaceuticals, and in 2015 delivered the Society of General Microbiology Colworth Prize Lecture. The Transient Expression system developed in his laboratory, that is CPMVHT, has been used worldwide and is the technology currently being utilized by leaf expression systems. His current research is particularly focused on plant-based production of viral particles for vaccine and diagnostic purposes. To know more about him, you can Google. Dr. Josh Lomonossoff is also a well-known figure in the field of virology. So enough intro, let's dig in. Let me give you a rough overview of today's seminar. First, our speaker, Prof Dr. Nicola J. Stonehouse, will talk about vaccine development using Vlp in great detail. Then today's chair, Dr. Abdullah Mamashwal, will greet you with his message. Then our honorable guests will join us in the discussion. In the meantime, our team will collect your questions and we'll go for a Q&A session shortly. So please make sure to leave your questions in the comment sections of Google Meet, Facebook, and YouTube Live ahead of time. Then we'll go for Q&A session round one. That is because Dr. Stonehouse has a very tight schedule, but we are really grateful to both of you for accepting our invitation and being here today. It means a lot. That's all from my end. Dr. Stonehouse, the floor is all yours. Dr. Stonehouse.
Thank you very much. It's great to be able to participate in this seminar, and it's wonderful to reach out to people in many areas across the world. So thank you very much for the invitation and thank you for putting this together. I think one of the positive things about the current world situation of coronavirus is that we have found some new ways of communicating, and this is one of them. So what I'd like to first do is tell you a little bit about the areas that my group work on. So we work on various aspects of the virus life cycle. We work on RNA virus replication, we have some work on structure, and we also work on VLP vaccine development. And it's that work that I want to talk to you about today. And we have a long-standing collaboration with Dave Rowlands, who's pictured here, who some of you might know. And also a lot of our projects have also been in collaboration with George Lomonossoff, who you're going to hear from in the second seminar. So you will hear some of the same themes, I think, in both the seminars today. So what I want to talk to you about are the following. I'm going to introduce to you what a virus-like particle is, and indeed I'll introduce what a virus is. I'm going to talk about why virus-like particles, we call them VLPs, are good vaccine candidates. And I'm going to talk also about a Vlp platform system to display foreign antigens. And this is the first example of the work that I'm going to talk about in VLPs. And this is the work predominantly of 3 PhD students, Kaniz, Jihad and Joe. Some of you might have met Kaniz when she visited the university in Bangladesh a couple of years ago now. And then I'm also going to talk about the second example, which are Vlp vaccines for polio. And this is work that was started by Palumi, Claire and Sam, and more recently has been, has been led by Lee, Sherry and Keith Graham here. And Lee and Keith also visited Bangladesh a couple of years ago now. And some of you might have met them. So vaccines. So vaccines, it's estimated, save between 2 and 3 million lives a year. And that's from the work of UNICEF. There are a number of vaccines which we all have when we're children, and many others which are optional. And together, we think these really are absolutely life-saving. Indeed, the combination of antibiotics, vaccines and clean water really are the most important things that have happened to improve health and well-being over the last years. However, there are still diseases that we don't have vaccines for, or indeed vaccines that could be improved, and that's what we're going to talk about over the next 40 minutes or so. So first of all, I'm going to introduce a little bit more about me. Thank you very much for the very comprehensive introduction earlier. I just want to point out where Leeds is. So this is our map of the UK. Leeds is over here, right in the sort of the centre, if you like, of the UK. George is over here. So this is where we are separated at the moment from our virtual homes. So Leeds used to be a very industrial city, and some of that industry is still remains around the university. It was famous for fabric, cloth production, and also dyeing, so colouring fabrics. But now the university is a mixture of old and new buildings right in the middle of the city.
So first I want to tell you a little bit about viruses. So viruses are basically everywhere. So every organism that we know about can be infected by a virus through bacteria, plants, animals, and indeed man. Viruses vary enormously in their shape, size, details of their life cycle. But what we're going to do is to try and strip back and just think about the very essence, the very simplicity of viruses. So what are the common themes? Well, they are all parasites. And in that, it means that until they get inside a host cell, and be that an E. coli cell or a plant cell or a human cell, they can't really do very much. They have to exploit the host cell in order to be able to undergo their life cycle and make more viruses. And in doing that, they often steal components from the host cell. So viruses that have membranes around them steal those membranes from the host cell. And this actually makes them very difficult to target, because it means they are in some part made or composed of some items of the cell. But at their simplest, they are basically a container and the genetic material. So these are some viruses here. They are shown by negative stain microscopy, when the stain stains the background, leaving the viruses as pale areas against this dark background. And this is about 25 nanometers in diameter. And at the simplest, what these viruses are, are containers. So I don't know if you can see my model here, which is a simple container, a little box, and inside is a wire, and that's what I'm calling the virus genome. The genetic material is inside, and it's protected and delivered by this container. So these are the things that people often think as the scary images of viruses, but actually these are inert. These are totally safe, and these don't cause you disease. It's the bit inside that causes disease. So it's the genetic material, when that's inside a cell, that's what gives you disease. So it's been estimated by colleagues at Sanofi Pasteur, one of the large vaccine manufacturing companies, that it can take $1 billion to develop a new vaccine, and there's got to be a real need and a real market. So lots and lots of vaccine candidates fail during development. It's a very expensive process, and there's got to be a real need. And I think we'd all probably have been, well, I think it's impossible that we've all missed the news in vaccines that have been coming out over the recent pandemic, and the real need and drive to develop new vaccines towards SARS-CoV-2, the cause of COVID-19. So how does the vaccine work? Well, to do that, I've got these three people. I've got a person who's naive, so who's never come into contact with a pathogen. I've got somebody who's recovered from infection or been vaccinated, and somebody who's infected. So in other words, you can become immune, or if, like this purple person, from having the infection and recovering from it, or from being vaccinated. So in order for a vaccine to work in this way, it's got to be a mimic, a really good mimic of the pathogen, or it's got to be a good mimic of part of the pathogen, as it possibly can be. And there's lots of ways of doing that. One way is to use inactivated viruses, for example, or inactivated pathogens, and that's a very classical way of making vaccines. And indeed, we still do that, and we're still developing vaccines that way. But virus-like particles are a much newer technology. And what that means, if we go back to our little container, is instead of making this and inactivating or killing it, you make just the container. So you never have the genetic material there in the first place. So therefore, your virus-like particle can never cause disease. And instead of making these in the normal cells, you would use a heterologous, a foreign cell system, so yeast, insect cells, and, as you'll be hearing later, plant cells. So these newer vaccines have still been around for a little while. Hepatitis B, where we're using the surface antigen, particles of that, has been around for about 20 years. The human papillomavirus vaccines, a little less time than that. So these are much newer. So because you're making only parts of the pathogen, and in this case, the container that the genome lives in, encased in, is a very safe process. So you don't need high quality, high containers to be able to do this. So it makes the process, in many cases, cheaper. So not just safer, but also cheaper. And for those of you who are interested in the human papillomavirus vaccines, these are the ones that are currently licensed in the UK. So the UK started using Cerevix and then moved over to Gardasil. And there's now new variants of this, Gardasil 9, for example. And these are by no means the only vaccines. There are other derivatives of these too. So what I want to talk to you about are two examples that we've been working on or contributing to in Leeds. And the first one is to talk about Vlp Platform Systems. So in this case, what we're doing is we're using a virus-like particle like this, and we are decorating it with antigens of interest. So we're not interested in the platform or the VLP itself. We're interested in this as a scaffold, as a way to display other antigens. And so in this case, this is our VLP. And these are, it's represented as a cartoon illustration as this blue ring. And we decorate the surface of it with antigens of interest. So the advantage of doing this is that if you display antigens on a VLP surface, you get a better immune response than just having soluble antigens on their own. And by having this scaffold that you can reuse, you can change the antigens. So you could make a mixed vaccine, for example, or be able to respond very quickly to a pandemic situation, such as we have at the moment. The other advantage is that you can make this scaffold and these antigens in two different systems. So you could make this in yeast, for example, very cheaply, or in plants. Again, very tractably, and you could then make your antigens in mammalian cells. And that would allow you to work with glycoproteins, which are challenging to make in some other systems. So you make your glycoproteins in mammalian cells, you make this in bulk, and you stick the two together. So a scaffold idea has been looked at by many groups, and the scaffolds that we're using are based on the core protein, also the capsid protein, known as the capsid protein from hepatitis B virus. And as I say, we can make these in a variety of cell types. So this protein spontaneously dimerizes, as shown here in this blue and red dimer structure, and then either 90 or 120 copies of the dimer come together to form a virus-like particle, as represented in the middle of the screen. And because you can get a varying number of these dimers coming together, this means you get a mixture of both T3 and T4 particles, with the T4 being slightly larger. And this is what they look like by negative stain EM. So we can put our antigens at two places on our VLP. So we can put them on the N-terminus of the dimer, and that will still end up on the surface of the particle. Or we can use these spikes that hopefully you can see running all the way around the particle. So this is the MIR, the major immunodominant region. And here is possibly the optimum place if you want to stimulate the immune response, to put foreign antigens all the way around this particle. But there's a problem. Because of the way that the dimer forms, we've got two loops here, which would be the insertion loops, and they're very close together. So if we insert in one, we'd get a steric clash in the other. So therefore, what we've designed, or rather what Dave Rowlands designed many years ago now, is a fused dimer, where the N-terminus of one monomer is fused to the C-terminus of another. So you almost make two proteins into one protein, meaning you can insert in one of these loops without putting anything into the other one. It means you get half the number of insertion sites, but you don't get steric clashes. So how do we put things onto this particle? Well, we are using a system called an affimer, and these are which are selected by the fixed part of the protein in green, and in blue there are some flexible loops which contain a variety of different sequences, and these are then selected by phage display to bind to a protein of interest. And because we want to generate a generic system, we want to have something that would mean we could attach a lot of different proteins, whichever sort of antigen we wanted. So we developed affimers that bind to a tag, and the affimers bind to a sumo tag. So sumo tags are used for protein purification. They're one of the tags that can be used, like a his tag or for example, and so this particular affimer will bind to the sumo tag with nanomolar affinity. So therefore it can trap, capture, and allow us to display proteins that have a sumo tag. So first of all, I'm going to show you some of the scaffolds, and we've got some nice high resolution structures of these. So these are, here we're looking at scaffolds that are made in Piscia yeast with fused dimers, each presenting an affimer to sumo at the MIR. And so you can see this quite nice scaffold structure. These are the larger particles here, the T equals 3 smaller particles. We can see very similar images if we also look at the scaffolds which have the affimer at the N-terminus. Here, each monomer is presenting the affimer at the N-terminus, and again, you're looking at the larger and smaller scaffolds. If we compare the two of them, the scaffolds, we can start to see a little bit of differences between them. So here, as our yeast-produced scaffold with our affimer at the MIR, and here our E. coli-produced scaffold with the affimer at the N-terminus. And what's quite clear, I think, is that there's extra density here. And this is density for nucleic acid. So here, this scaffold has packaged nucleic acid. This one hasn't. At the moment, we don't know whether this is because this is in yeast and this is in E. coli, or whether it's because of the different constructs. That work is ongoing. But also what you can see, hopefully, is you're getting this extra density all around this N-terminus scaffold. So we're getting a lot of more detail about the affimer. And if we change the contour level, as I've just done, it's now much clearer that we're really seeing much better density for the affimer with the N-terminus structures. So what this could be showing us is that this is a more stable, less movable, mobile structure. This is a lot more mobile, and therefore when we are not able to see the affimer in such detail if it's a lot more flexible. So the next thing is, do they work? Can the VLPs actually capture antigens of interest? So we've done a number of different experiments here over the years, both in George's lab and in Leeds. And I'm going to show you just one piece of data on some of the development work. So here what we've been trying to, what we're saying is, if we make a foreign antigen and take one of those scaffolds that I've just described, do we actually manage to immobilize that antigen onto our scaffold? And we've used the glycoprotein, a glycoprotein that's come from Hunin virus. This is an arenavirus. It causes Argentine hemorrhagic fever, and there isn't a vaccine currently. So this is one of the diseases where there is a need for a new vaccine, for any kind of vaccine. And the glycoprotein fragment has been produced by our colleagues in Oxford, Tom Bowden in Oxford, and he made that in mammalian cells, in 2932 cells. And it's heavily glycosylated. So we've got different ways of being able to monitor how VLPs are decorated. And I'm showing you here some data using sucrose density gradients. So because the VLPs, will sediment into the middle of a 5 to 15 to 45% sucrose gradient, and we can detect them by Western blotting. If we use decorated VLPs, and detect for the antigen, if this actually co-migrates, which is happening here, this shows us that we've actually decorated the VLP with the antigen, and this is interaction is stable enough to persist through the sucrose gradient. If we use scaffolds that haven't got an affimer, so they can't bind the antigen, then we still detect the scaffold, but the antigen stays at the top of the gradient. So that's looking very promising. So what did we want to do next? So if we go back to March, what we wanted to do was to take our VLPs that we had already characterized and already had our nice structures and decorate them with the receptor binding domain from SARS-CoV-2. And we plan to make this receptor binding domain in two different cell types, in mammalian cells or in yeast. And just to orientate you to what I'm talking about, this is a cartoon of SARS-CoV-2, and the spike is in yellow, and the receptor binding domain is at the end of the spike, and we think this is one of the major antigenic determinants, and so this is an ideal vaccine candidate. So instead of this decorating SARS-CoV-2, let's decorate our VLPs with it and see if we can make a synthetic vaccine. So the labs in Leeds unfortunately closed, but our colleagues at Oxford were allowed to work and able to work in a very limited capacity. And they made the receptor binding domain with a SUMO tag. And you can see that protein here. It looks nice and pure. They then mixed the VLPs with the RBD SUMO and took some images. Here is you're looking at these in vitreous ice. So if you're not used to looking at images in ice, you can't see a lot of contrast intentionally. You see a lot less contrast than negative stain, but you can hopefully see that you're starting to get sort of slightly dotty haloed particles, which gives you an indication that maybe they're decorated with antigen. And we did this both for the constructs made in yeast, the fused dimers, and the N-terminal constructs, which are made in E. coli. But I should also point out at this stage we don't have an awful lot of data. We have more data for the MIR, and this is really very preliminary work. However, if we now are just looking at a little snapshot, a little part scaffold, now we can start to see, if we change the contour a little bit, some extra density here, which corresponds to the RBD, which looks as though it's been captured and sitting here at the N-terminus. The data for constructs where we're capturing the MIR is much better, but we have much more data thus far. So here we can be a little bit more confident that what we're seeing is density that corresponds to the RBD. And we've got some very initial binding data, which has been done by, again, colleagues at Oxford, to say that our scaffolds are capturing the RBD, and the affinity is around about 40 nanomolar, which is nice. So scaffolds can be very useful. And I've shown you a flexible antigen presentation platform that's where we can make that in either E. coli or Pichia. And we also know we can make that in plant cells. And this could be used, I think, in two ways. We could use it as a potential vaccine candidate, but we could also use it as a way to present antigens for ELISA. So we need to detect antibody responses in patients in clinical trials, for particularly for all the COVID trials that are going on, and to monitor what happens for recovering patients, for example. And so we're in the process of evaluating this with colleagues in Oxford. So scaffolds are good, and scaffolds can be very useful, but scaffolds aren't the answer to everything. There never is often one answer to everything. Is there another way that VLPs can be useful if you have complex antigens? So in other words, if you've got a situation, and we go back to our little model, where your main antigenic sites are not just sitting nicely on one protein, they actually straddle protein-protein interfaces. And in that situation, a scaffold system really isn't very useful. And here we come to our VLP vaccines for polio example. And so this is a consortium project. It's been running for about 10 years now, and it involves the University of Leeds. It's leading the project, also involves the John Innes Centre, led by George, the National Institute of Biological Standards and Control, led by Andy McAdam. These people do batch release of vaccines. The Pirbright Institute and Toby Tuthill, University of Reading, Ian Jones, University of Oxford, Dave Stewart and Liz Fry, and recently University of Florida, Flanagan has joined the consortium. So this was us at the beginning of our work. So as I say, this has been ongoing for about 10 years now, and you can see that George is there, I'm there, and we're looking quite fresh-faced. This was last year. Everybody's looking considerably older, and you can't even make out George on this because he wasn't unfortunately able to attend this meeting. So I want to tell you a little bit about polio before I go on to talk about some of the data. So what we've been trying to do, why polio is still a problem, and why we need a new vaccine. So most people don't know they have polio. It can just cause a very mild illness that people fully recover from. But for some people, it can cause paralysis, and for some, this paralysis can be permanent and can even be fatal. It's highly transmissible because it's through water, through food, through ingesting contaminated water or food. So it passes from person to person very, very rapidly. And there are 3 serotypes, and that's important as we look forward, because it means we don't have to just, making one vaccine candidate isn't going to help us. We actually have to make a mixture of 3. And if we go back to the pre-1960s, polio was the primary cause of disability worldwide. It still is a cause of disability, but a much, much lesser case than in if we go back 50, 60 years or so. This is poliovirus. It's at relatively low resolution, so I can get it to whir around quite nicely, and it's a complex virus. So the virus capsid, although at first glance just looks like yet another one of those containers you've seen before, but here the capsid is made of 3 different proteins, and the 3 different colours, the red, the green and the blue, are 3 different proteins that have to come together to form this capsid. So vaccination has been incredibly important in controlling polio, and if we just look from the 1980s through to a few years ago, the number of cases have come down enormously. So this looks great. This looks really, really encouraging. But unfortunately, the cases haven't gone to zero. The devil's in the detail around here, where there are still significant numbers of cases. So why is this? Well, we still have outbreaks of polio from wild-type virus. And this is predominantly now in Afghanistan and Pakistan. Although, of course, people move and travel, and therefore you will get outbreaks spreading around. So Nigeria very recently has been classified as polio-free. It was during the COVID peak of the COVID pandemic, so it was kind of lost in the news, I think. Also, there are unfortunately cases of vaccine-derived polio from the use of the oral vaccine. So the oral vaccine, which is an attenuated virus, has been responsible really for all of these decrease in polio cases that we've seen here. The success in reducing these cases is really because of using the oral vaccine, but it has a downside. And this is because it is a live virus, and that live virus can mutate and it can regain the ability to cause disease. And that happens. It's rare, but it does happen. And what that means is basically that the vaccine that you take gives you polio. And here, these figures are quite shocking, I think, where we're looking at the number of polio cases worldwide. In 2018, we had 13 from wild-type virus and 104 that came from the vaccine. So in terms of continuing to use the live vaccine, this means that it is really untenable. We can't carry on with there being more cases of paralytic polio worldwide that have been caused by the vaccine. So many countries now are moving to the inactivated vaccine. So this is where the vaccines are prepared in quantity and they're treated with formaldehyde in order to inactivate the virus. And so going back to our model, what you do is you make lots and lots of virus, and then you basically still got the genome inside, but you cross-link it. And so by cross-linking it, it means that that virus is basically all locked together and can never infect a cell because the genetic material never get out. So that's very safe in order to use, but it's not necessarily safe to make because you have to have loads and loads of cells all infected with virus in order to have this approach. And that's not great going forward as we move towards a virus-free world, to keep making virus just to create a vaccine. So why do we need to keep vaccinating if we're going to eradicate polio? Well, that's because of long-term poliovirus excretors. And these are people who've had the poliovirus vaccine, the oral vaccine, but haven't cleared infection. So these people are immunocompromised usually, and they continue to excrete polio, vaccine-derived polio, for years and years in some cases. And so one of these people is all you need to basically repopulate the world with poliovirus. So for the foreseeable future, the WHO, we do not cease polio vaccination. And that's where we came in, with a safer way to make a vaccine. So I need to explain to you about the poliovirus genome. So this is the genome. It is a positive sense RNA virus. It has an untranslated region at the end, and then all of this bit in the middle, the genetic sequences, what happens is this is translated directly into protein when the RNA gets inside a cell. So when translated, it's also cleaved up. So it's a polyprotein that's cleaved up into the structural regions here in green, non-structural regions in violet. blue. And these include proteases such as this 3CD and this 3C, which are involved in cutting up this polyprotein and are also important in our vaccine strategy. So I need to explain to you how the virus normally assembles inside a cell. And if you remember, there were those three colours that you saw earlier on, and those three colours were three different proteins. So normally what happens is you have assembly, maturation, cleavage, and you get production of our nice virus, which has its genome inside. If there's no genome around, you can get empty particles. These empty particles look exactly the same as the virus. So this is almost like the virus creating virus-like particles for us. The virus has done the job for us and made Vlps. Fantastic. However, the downside is that these are not very stable. And this is shown, I think, best by this image here, where you can see the structure at relatively low resolution, 8 angstroms, of native and non-native particles. So this native particle, a little smooth around the edge. This non-native is quite angular, and these five-fold regions are really quite protruded. And you've also got this these gaps here because this particle has slightly expanded. So what happens when this occurs is you've lost the normal antigenic sites, and as a result of this particle doesn't elicit the proper immune responses, as does our native particle over here. So although we can produce empty particles, they are no use at all as a vaccine. We have to stabilize them. We have to make sure they stay like this and don't pop out like this. So what I'm going to do now is summarize an enormous amount of work over many years, and the work, as I say, of the consortium and many people in the consortium. So what we did was to try and stabilize the particles, and we introduced a large number of mutations. There's a pocket region in the capsid, which has a lipid. We tried to stabilize that, again by mutation. And we thought, we know a lot about structure, we can do this. And unfortunately, most of these approaches were not successful. What we then did was use an evolutionary approach. A number of which is where we took a virus, expressed it in some way. And in this way, we generated mutations which stabilized the capsid, stopped that shift in conformation, made the capsid more stable, more rigid. And those mutations were then the ones that we wanted to go and move forward in. So at the moment, we're still working with virus. But obviously, if we want to make a VLP, we need to start translating into different cell types. So we want to be able to make our empty capsids in a heterologous system. And we used a number of different systems. We've used insect cells, we've used plant cells, as you'll hear about later. And what we've been focusing on in Leeds is yeast, particularly Piscia. But all of these approaches use the same technology, where we are taking the structural protein region, this green part here. We are co-expressing a protease, and that allows the protease to cut up these proteins into their three separate parts and assemble a VLP. So it all sounds quite straightforward. In practice, of course, it was quite challenging. What we use in Leeds, as I say, we've been doing a lot of work with Piscia pastoris, or Lee Sherry and Keith Graham have particularly, and this is the cloning strategy they use. We've got a commercial Piscia Pink system where we're using an alcohol oxidase promoter to express here the structural proteins and here the protease together. And here are some of the VLPs. So this is back to negative stain EM now, and you can see these VLPs now looking quite pretty. So how stable are they? Well, to look at stability, we look at the level of native antigenicity, which we call de-antigenicity, over temperature. And we compare this to the current vaccine. So bear in mind that the current vaccine, the IPV, is also cross-linked, so is going to be particularly stable. But over time, you can see that we are, over temperature, we're able to stabilize our particles pretty well by our combinations of mutations. Here, these have 6 mutations. This one has 8 mutations. And these are the 3 different types of polio, PV1, PV2, PV3, 3 different serotypes. So the idea isn't to generate something that will necessarily survive 60 degrees, but we're using temperature here as an idea that, yes, we're creating something that's more stable, because without these mutations, the stability is, the antigenic stability is lost even at 10 degrees. And then what we want to do is to say, well, what happens when we put these into an animal system? So we can use a rat system where we can basically look for the production of neutralizing antibodies. And we can also use a transgenic mouse system, which where we've got mice that express the poliovirus receptor. And these mice can then be immunized with the sample, boosted with the second immunization, and then challenged with wild-type virus to see whether or not they get disease. So in other words, is this vaccine candidate protective? And obviously we need to do this with all our 3 serotypes. And this work is done at MIPS. So I'm not going to show you all the data. I'm just going to give you a little bit of flavour of the data to show you that things are looking pretty positive. So if we look at PV2, which is one of the serotypes, now we can see and look at the antibody levels. With 1 dose of IPV, 2 doses of IPV goes up, as you would expect. And with our Vlp made in yeast, beg your pardon, you get, again, an increase in antibody titer. And this antibody level titer is pretty similar to what we're getting with IPV. So that's looking good. What about protection? Well, if we have 2 doses of VLP, all of the mice survive. And that's really good. And that's the same situation also as shown here, and also the same with the IPV. But you did need 2 doses. For the PV1 here, we're getting quite a good response, even with a single dose, which is still enhanced with 2 doses. And here, all the mice are surviving with a single dose of VLP, which is really quite nice to see. So I'm going to leave you with the structure of PV1 VLP at 3.3 angstroms. It's done by colleagues at University of Oxford. Hopefully you can see this looks nice, smooth, doesn't look expanded. So not only do these VLPs look right, but they also elicit the right immune response. We've still got some challenges to come. And one of the challenges that we are working on is upscaling. So making more of something isn't necessarily straightforward, and that's something that we really are working on at the moment. The other things are there are differences between some of the VLPs, then the yields are different and some of the characteristics are different, and we need to understand that a little bit more detail. However, I've shown you that we have been able to stabilize the polio capsids, and that these are have a lot of properties that are similar to the current vaccine antivirus. So they're looking very promising as a vaccine candidate. And as WHO has already put out an expression of interest for people to work with us, companies and CROs to work with us. This got interrupted by the current pandemic, but they're going to issue that again. So hopefully we can move forward with upscaling. Unfortunately, the polio eradication campaign also got halted through the COVID pandemic. So we even more are going to need a new vaccine, because there is likely to be an increase in polio cases, I think, as a result of that. And I'm going to leave you with this. Although yeast is a good way to make Vlps, it's not the only way. And George is going to take that idea forward and tell you about sort of alternative ways of making Vlps in plants. And I think everybody who is involved in vaccines knows and understands that there's never one way of doing something. And indeed, if you want to vaccinate the whole population of the world, you can't do that with a single approach. You've got to have multiple approaches for whatever vaccine you use. So together we are stronger, I think, is what I would say. And I'll leave you with that. Thank you.
Thank you to the Stonehouse. That was a very captivating presentation. I have learned a lot from you. We're lucky to have you with us. Thank you once again. And now I'd like to request our event chair, Dr. Abdullah Muhammad Shralsar, to share a few words with us about this new platform that he has started and his vision with it. Dr. Shralsar.
Thank you, Saim, for hosting this event. Assalamu alaikum. Eid Mubarak. Already Sam Hospital explained who didn't know about the Eid Mubarak. First of all, I'd like to thank from core of my heart Nicola Stonehouse and George Lomonossoff for our invitation, and then give their valuable time to basically, I would like to say, teach our students for curious mind, or teach our fellows. Nick is our old friend. She visited. I hope George will also be visit here, and we'll have the sons working together. I am not talking much because Nick already very nicely described about Vlp applications, host, and everything. But still, we have a few questions to Nick. Our students ask those questions. So before going the question, we will have very short discussion, and I would like to invite Dr. Mainul Ahsan from Incepta Vaccine, simply working on Vlps in Incepta. So please, after your some question to me. Dr. Mainul Ahsan.
Hi. Good afternoon, Nick. This is Mainul. Can you hear me?
I can. Hello.
Hello. Hi, good to see you here. Yeah. So
Yeah, very
basically,
good. Very good to hear you.
Yeah. So I'd like to say one thing. Nick is very good friend of Incepta. We know each other since last 5 years, more than 5 years, I guess.
Mm-hmm. Yes.
You know, and she has also done some work for us for electron microscopy for our HepB project. So basically, you know, Nick, you know, we are working. You know, we have a good platform, yeast-based platform for Vlp. So we already developed a product, Hepatitis B Vlp. This in using the PCR system. The vaccine is in the local market already. Okay, and you have done the transmission electron microscopy for that one. So, but we are also working recently, since last 2 years, another 2 product, which actually I don't want to disclose the name in this platform. So basically, from your scaffold system, it's very actually a nice system and very promising. From what I understand, you have already made the RBD, you know, with this scaffold system, which is presenting the RBD from SARS-CoV-2. So have you done any preliminary experiment showing that this scaffold system presenting RBD is immunogenic? I mean, in animal study, have you found any data?
So the RBD was made in Oxford. I should also make that clear. So we made the scaffolds and they made the RBD. We haven't done any animal experiments yet. It's been so difficult to carry on working during the shutdown period here in the UK. But it is very much on plan. So we are back in the lab now, and we are making more scaffolds. So yes, it will be very much on our priority list to look at immune responses.
Okay. Thank you. We are looking forward to seeing the data. So basically, for your polio Vlp, is it self-assembled or you did some sort of chemical treatment to facilitate the assembly?
Can you say that again? The polio VLP?
Is it self-assembled or you have done any? You have. I think you understand the question.
Yeah. Okay. Yes. Yes. Also, it's all self-assemble. There's no chemical cross-linking or anything like that. It's whatever system that we use. If we co-express the precursor protein for the structural proteins and the protease, then we get self-assembly. Actually, we're using we're using different constructs, obviously for different systems, but it's all for all of those systems. It's self-assembly.
Okay. So basically, during the development of polio Vlp, sometimes what happened during the assembly of the Vlp, some unusual protein from the media component or DNA on each protein may get enter into the Vlp. So have you faced any sort of problem like that?
So in terms of
if so, how did you manage to overcome?
So yeah. So I think the question then was, are there any sort of foreign contaminants in the VLPs? So in terms of inside the VLPs, as far as we can ascertain, we don't have nucleic acid contamination. So we can't be sure that there aren't sort of small nucleic acid fragments, but there seems no discernible nucleic acid, so not a lot of nucleic acid packaged. I mentioned very briefly about a pocket in the surface of the VLP, and this is sort of invagination, if you like, into one of the proteins. And in that pocket is normally a lipid, and that's picked up from the host cell. So in the yeast VLPs, there is a lipid there, but it looks like it's a much shorter lipid than we get in normal virus. So that could be considered a contaminant, I guess, but is important for stability of the pocket region, we think. And we could also think about replacing that with other compounds, other drugs, for example. At the moment, I don't think our VLPs that we're making are really pure enough to be able to say if we get small amounts of other contaminants. I think so. That could well be that we've got other things that are either inside or bound onto the surface, and I don't think we're at the stage of really knowing that yet. I think that's something to move in the future.
Yeah, thank you. So basically, why I'm asking this question, because there is a recent paper about the influenza VLP. In that paper, they found something inside there. Okay, now actually in the vaccine development, the regulator don't know about it. So people in future they might some sort of regulation to check this. Okay. Yeah.
Yeah. So we have spoke. Sorry, we've been speaking to people who are sort of mass spectrometry experts as well to see whether mass spectrometry is going to be a way also of looking at contaminants going forward. But as I say, I think we're at the moment our preps aren't pure enough, I don't think, to be sure of what's just co-purifying rather than what's actually bound, and that's the difference. Yeah.
So I have another question. During the development, suppose you have in the supernatant you have got the good VLP, which is auto-assembled. But in some approach, people use deassemble then reassemble.
Yes.
Which have you done anything like that? And if so, and why?
So we have done disassemble, and we have done some disassembled and reassembled for the Hep B, the scaffolds, the Hep B core protein. You can disassemble and reassemble those. Yields go down quite a bit, and the reason that we did it was it's a way of getting rid of nucleic acid from the inside. So for the VLPs made in E. coli, where they have nucleic acid inside, if you want to get rid of that, you can disassemble and reassemble. However, for the polio VLPs, we have done a little bit of work on disassembly and reassembly, but I wouldn't say we haven't really done very much on that at all. Certainly once you've had a shift from non-native from native to non-native, you can't go back. That's a route that's irreversible. So once the particles have expanded, you can't go back on that. But there is some evidence that you can disassemble the native particles and put them back together. But we haven't got any real rationale for doing it. I know it is done for the part of the purification of some of the human papillomavirus VLPs, though. But just to add, you mentioned finding the VLPs in the media. One thing I didn't say, and I should have said, is that all of our VLPs that we're making in yeast are not secreted. We actually have to lyse the yeast. So they're not secreted. They are, we haven't put a lot of effort, but we haven't had really any success in secreting.
All right, yeah. So basically you're talking, actually the de-assembly or reassembly could increase the stability or an immunogenicity. What do you think?
I mean, I think it could improve or reduce heterogeneity. So if you reduce heterogeneity, you could improve the immunogenicity, maybe. But I think it can be very costly to do it because certainly for the Hep B cause, we lost a lot of material. So it actually might, for us, I don't think it would be necessarily worthwhile to do that.
Okay, so I have a last question just from my work. Actually, what I am facing, I have after purification, I am getting good VLP. Okay, good yield is good. I can see the one peak in the HPLC. Also, I can measure the protein concentration by BCA and Lorry. But when I run that protein into the SGS space, in denaturing condition, there is nothing in there. Any idea?
So you've got a peak on
HPLC,
HPLC, and you've got.
I can measure the concentration by Lorry or BCA. Okay.
So you can do a BCA assay and you can measure the concentration, but when you run on STS page, you don't get anything.
In denaturing condition, nothing.
Not even stuck in the wells. Not even. It's not stuck in the wells.
No, not even not in this. Okay.
George, any ideas? You're muted.
He's not.
He's muted. I think. Yeah.
George, please unmute.
So can you just say that again? Just the question. Didn't quite catch.
So hello. Hi.
I'll paraphrase the question, George. So this is Maynard was asking about his own experience. He has a protein a VLP that he's getting. He can measure concentration with a BCA type assay. So it looks like he's got material and protein there, but he sees nothing on an STS page.
Well, that's yes. I mean that's interesting. Of course, some proteins don't stain very well.
Yeah.
Of course, depends on what stain you use. I don't know what that would be, and the sensitivity. I mean, you can use either things like Kumasi Instant Blue, or of course, things like silver stain are more sensitive.
There's an orange one, isn't there? There's an orange something.
Yes.
It's an orange stain. I can't remember what it's called.
Yeah,
which sometimes works.
Of course, one of the things can happen is with VLPs, they can get cross-linked, and so everything stays in the well, so you don't see the individual subunits. And we've certainly had that problem. So if it's a very stable VLP, that can sometimes be an issue to actually break them apart efficiently. And one of the things we've also found is that this kind of tendency to stay in the wells is often worse under reducing conditions. If you include dithiothiazole or mercaptomethanol, that often causes this tendency to stay in the wells rather more severely, which to me is counterintuitive. I thought it would be better at breaking things down, but it just seems to be an effect. So sometimes you will just see things sticking at the top of the gel. I don't know if that's what you've seen or. I can't quite.
Okay. Yeah. Okay. Thank you, George. Yeah.
Didn't quite catch. So I didn't quite catch it the first time round. Okay.
Okay.
Okay, Nick, thank you.
Manu, thank you. I have a question. Actually, not question. I am asking you to give some comments about this. You are working on Vlp in Incepta vaccine. Sorry, Incepta RNB. So, would you please tell us about something that how easier working on Vlps in Bangladesh like other labs or institutes or everywhere? Because lots of students and researchers are gearing up and they are very much eager to work on that. So if you would just tell us about how easier to work with
VLPs. So I mean, when we came, Lee and Keith and came to set up the practical classes, which they ran in Jahangir University, that was working with hepatitis B Vlps that had been made in E. coli. So making the Vlps in E. coli can be achievable. You can also make the Vlp with a his tag, meaning you can use affinity purification or beads, which can make purification relatively easy. So, so E. coli purification does work. Yeah.
Okay.
So I think, to go back to those ideas of the practical classes, the master's classes that Lee and Keith set up a couple of years ago. Yeah.
Yes. Yeah, I'm going to Mainul again. Mainul, you are working on Vlps, and how do you recommend working Vlps with other labs or institutes?
Oh, everything's gone. Ooh.
Please say that again, please.
What do you think? I mean, from your experience working on Vlps, should we go in different institutes or universities or labs working in different aspects of Vlps?
So should we go to different, you said go to different institutes, go to different labs?
Yes, Nick, actually only Incepta vaccine is working on Vlps here, but other institutes and other universities, we think that is very difficult and it's a very, I mean, we need sophisticated instrumentation or we need lots of fundings, lots of constraints we do have. So actually, we are not confident working. So that I'm asking Mainul, as he is working in Incepta Vaccine Limited there. So if he has any recommendation to the audience.
Yeah. Okay. Thank you, Sohail bhai. Yeah. Basically, initially, in the Incepta, we have already developed a yeast expression system. We can now express Vlp both intracellularly also extracellularly. So basically for the Vlp development, the most important things is first the molecular biology part, the design part is very important. If something goes wrong with something wrong in the design part, then you have to face a lot of problems. So still, I wouldn't say this is very difficult, but I rather I would say it's very challenging. What do you think, Nick?
Yes, yeah, but that part of the design process, of course, that can be done in university labs. There's a lot of things that you can't do in university labs, that's sort of the upscaling and things like that are very challenging. But I think doing some molecular biology is possible. And I think we showed that in the practicals in Jahangir that some things were possible, not everything, but some things were possible.
Dr. Nick, how much cost is involved? That's what Sarah was asking. So how much cost is? Yeah.
So our main costs, I think, are staff costs. Cost of employing people is one of the major things. So that's obviously animal costs. If you do immunogenicity experiments, can start costing quite a bit of money.
Just a rough overview.
Media. I beg your pardon?
Just a rough overview of how much money do we need to start working on this Vlps?
I mean, just to do simple expression and purification of it, a little bit of cloning, expression and purification, you're probably looking at sort of hundreds of pounds, not thousands of pounds. It's having hands to do the work, and some of the equipment should be reasonably obtainable. But I understand the constraints and the difficulties. But if you make Vlps in E. coli, it's much easier and cheaper. Yeast need expensive media, more expensive media, and it's much harder to work with yeast. But E. coli is still useful, so you can still make Vlps in E. coli and grow E. coli. Is an easier thing to do. So that's what I would suggest.
Okay, thank you, Nick. I think everybody understood already. So a few questions from the audience. So George, you have special question after your lecture, but if you want to help Nick, you can also add some values on Nick's question. So right now I'm asking this question, to Nick. Number one, someone asked how a vaccine maker starts his or her journey for being vaccine scientist. What are the barriers for vaccine development?
So I didn't catch all that. So there's a journey for vaccine scientists, how to do that?
How to become a vaccine scientist.
How to become a vaccine scientist. Okay, I guess I don't think I am a vaccine scientist, really. I think I'm someone who uses molecular biology, biotechnology, and virology to answer questions. So I would say, get a good grounding and a good understanding in molecular biology and biochemistry, biotechnology, and that's the good first step.
Yeah, if I could just add to that, my background was originally in plant viruses, which was a million miles from vaccines.
Yeah.
And so I'm always looking back, I slightly look back on amazement at where I am.
Yeah.
But it does show you that you can actually start in a different place.
Yeah.
But I did work on virus particles in a days when a plant virus was particularly useful because of the amounts you could get. It's actually, believe it or not, it was pre-cloning. With that background, I could then, try and adapt what I learned to go into projects like the one on polio funded by the WHO, which is not, I imagine, when I started. So, I would just reiterate, it's knowing the basics of molecular biology, cloning, expression, and some understanding of some aspects of structural biology, and even understanding particle structure. I don't think necessarily to get started you'd even need to know an awful lot of virology about how the virus infects. You just need to know that those parts have been identified and have means of expressing them in whatever system you choose. And then have a means of assessing what you've made. And another thing I would say is it's very important to partner people. So, for instance, I work at a plant institute with the polio project. There's no way I could test anything with live polio. So we rely very much on our partners, particularly in London, the National Institute of Biological Standards and Control, who can handle live polio. Likewise, it's something I'll mention briefly, and that Nick has already mentioned, the structural work with cryo-electron microscopy through collaboration with Oxford. And so it's an important thing is to establish the collaborations and to have a good network.
Okay, I think our participants got a very good answer and then very positive answer. Who are thinking to become vaccine scientist. Next question to Nick, why virus like particle? I mean, why Vlp? If you say, you already answered in your lecture, but if you please just in a single sentence, if you answer, why Vlps?
Why VLPs? They are safe and they are cheaper.
Okay, that's a good answer.
Okay, can I also add that all recombinant vaccines which have been on the market, and that's not a huge number, are based on VLPs, because rather than soluble proteins, because of their size, they're recognized very well by the immune system. They are recognized as being similar to the pathogen in a way that soluble proteins aren't, and they can be taken up by dendritic cells. And so VLPs are very good mimics in structure of the pathogen, and that helps them to give an authentic immune response. You can always raise response against the soluble proteins, and we often make reagents for serology, but VLPs are the ones which will actually tend to raise the best neutralizing protective response.
Dr. George, can VLPs replace native viruses?
Oh, sorry, I didn't quite catch that.
Okay. So can VLPs replace native viruses?
As a source of vaccines,
Yep.
Yes. I mean, I think they will do. We've heard from the Poliovirus stuff, and they're great because they're non-infectious. So you can work with them much more easily, and they're very, very safe. They can't revert, and we've heard from Nick about the problem of reversion of the all-Sabin poliovirus causing paralytic polio. That can't happen with a VLP. It's never seen any genetic material, well, not infectious genetic material. So even in a plant institute, we can make poliovirus VLPs. There's no, danger from that point of view. We don't have to work under category 3 conditions. We can work on the bench. These are just they're protein shells, essentially. And so that's why there's such a huge interest. The other great thing about them, there are quite a few viruses, and human papillomavirus was a very good example, which Nick mentioned, is it's very, very difficult to culture the virus. And I mean, in practical terms, next to impossible. But you can actually produce, using molecular biology techniques, VLPs quite readily. So you cut out the need to have to culture the virus, which can be extremely difficult in certain cases.
Okay, already one question answered, so I am going to the next question. It was discovered in 1983. Why still now there is no effective vaccine against HIV? So everybody is worried. What will happen about COVID? Coronavirus vaccine.
So I think the situation between HIV and COVID are very different. So the viruses infect in different ways and present different challenges to vaccinology. The amount of money that's being spent towards a COVID vaccine is phenomenal. There are rising 200 different vaccine candidates now in some sort of trials, animal or clinical trials. So the enthusiasm for a COVID vaccine is vast. It still might not work. There are challenges. Eliciting an immune response from the vaccine seems difficult, but yet some of the cell responses look good. So I don't think. I think we've got a good chance of getting a COVID vaccine. The question is when.
Yeah. One of the questions to add to one of the problems with HIV, of course, is that the virus itself attacks the immune system. And so it's extremely good at hiding from it, because the immune system is the target in that case. And there is a cautionary tale there that you're going back to the 1980s, there was a lot of excitement about various epitopes on the surface of HIV, certain protein sequences, which would actually raise very strong antibody responses, which is the first thing you look for in a vaccine. But none of those, the presence of those antibodies did not confer protective immunity. So simply getting a good antigen response is not a guarantee of protection.
Yeah.
One advantage we also have of COVID-19 is that although coronaviruses have not really been considered major human pathogens, well, certainly not until SARS in 2002, they are very important veterinary pathogens, particularly of pigs. So there has been a lot of background work on the molecular biology of coronaviruses, mainly aimed at veterinary vaccines. And so we are basing it on quite a lot of knowledge of the fundamentals of related coronaviruses. It's not that that class of virus is new. I mean, they were first described actually as long ago as the 1930s. But most people haven't really been aware of them because they've not been of interest in human disease particularly. I mean, they're associated with colds in the past, but they've really been of veterinary importance.
Another question, how much reliable are those Vlps regarding the stimulation of human immune system against polio and other emerging diseases?
So you won't get the same immune response that you get to live polio, because polio elicits a mucosal immune response, and VLPs are very unlikely to do that. So you're, in this case, you're not going to be able to have a perfect mimic, but hopefully it's good enough. So certainly and what at the moment we're not trying to mimic live virus, we're trying to mimic IPV, the inactivated vaccine, and it looks as though we are able to make a good mimic of that. So I think it might not be perfect, but it might be good enough.
Just to add to that, there is evidence that having RNA, even if it's not infectious RNA, within a particle can mediate a TLR response, which can potentiate the immune system. The other thing you would have, say, is either with a killed vaccine like the Salt vaccine, and certainly with a live attenuated, you would also get the presence to a certain degree of non-structural proteins, which could contribute. Whereas with the VLP, you're only going to make the structural proteins. I mean, you could deliberately try and engineer some non-structural components. So in a sense, they have a rather more limited set of epitopes, but you hope they have at least the major ones, which could actually stimulate infection, immunity against infection.
Yeah, so it's kind of you've got to make some sacrifices, I suppose, in order to be able to make something practical and safe. And so you focus on the most important bits, and you don't worry about the less important bits at the moment, but maybe we could add those on later, as George says. Yeah.
Among many questions, this is the last question from the participants to me. One wanted to know about zoonotic VLP.
Zoonotic VLPs.
Yeah.
So is that, you mean VLPs that you could use to treat animal infections rather than necessarily human?
Yes.
Yeah. Okay. So certainly that's the case. There's a really good potential market for treating animals, and the other advantage is you don't have to pass the same kind of safety constraints as you do for humans. So George mentioned the possibility of having nucleic acid inside VLPs, and that can be very stimulatory to the immune response. So that's really good. But for human vaccines, that's really tightly controlled. It's not so well controlled. It's not so tightly controlled for animal vaccines. So that can be a big advantage with producing vaccines. Yes. So yes, indeed, VLPs for animals. Yes.
In fact, I'm going to talk about, if we have time for my presentation.
Sorry, George.
No, that's fine. No, it's a discussion. But I realise other people are getting late in Bangladesh at the moment, so I don't want to, if there isn't time, maybe another time we'll do it. But the first two examples will be actually veterinary vaccines, and that's for the very good reason that Nick said it's much easier to test them than. But also, yes, you could certainly. I mentioned coronaviruses. A lot of the work on making VLPs a coronavirus was initially done for veterinary purposes, and coronaviruses, including the one that's responsible for COVID-19, is definitely zoonotic.
It's a zoonotic virus.
Yeah, it's definitely, and there are plenty of others, emerging viruses, which are like that. I mean, a classic one of recent years is Ebola as well. And that's a big problem if you have an animal reservoir, particularly wild animals. Even if you have the most efficient vaccination program for humans, you're never going to vaccinate all the wild animals in the reservoir. So it'll always return if you let the vaccination rates drop. It's there, it's always going to be circulating in the animal population. So that's an important thing to remember, I think.
Yeah.
Okay. Thank you so much, Nick, for your very nice presentation and answering the questions. And we will be in touch with you if some more questions from the audience has come. So I'm requesting Simon to come back further.
But so my pleasure.
Thank you, Dr. Nick, for being here today. And you were in our university. You visited Jahangirnagar University when I was up there. And I hope you will come again in future. So thank you, Dr. Nick. Now I will request our honorable speaker, Dr. George Lomonossoff, to provide a comprehensive overview of vaccine development using transgenic lens. So, George.
Okay, this is going to be something of a tag team with Nick. So we are a well-oiled machine, and so we have colluded in our presentations. I have to admit, to make sure we didn't do exactly the same thing. So I'm now going to do my presentation. I hope this all works fine. Let's see how we go. Can you see that?
You are not presenting.
Okay.
Not yet.
Not yet. Let's see what's happening here. It works so perfectly in sharing.
In our test drive, yeah. So I guess Nick will have to stop the presentation. So if Nick is presenting, then first Nick will have to stop the presentation, and then Judge Lomonossoff can present. So I'll request Dr. Nick to stop the presentation if she's presenting. So I guess Dr. Nick is not presenting right now, so I think you should give a try.
Is that
yes,
okay?
Yes, it's working perfectly.
I better start at the beginning. I don't want to give away the outcome, the plot. Okay, can everyone see that?
Yes, we can see that.
Okay, well, first of all, I'd like to thank everyone for giving me the chance to present some of the work in my lab. And the picture you see at the front is actually an aerial view of the place I work. And I hope you can see the cursor. The building I actually work in is this rather cubic looking building here, which is the so-called chat building. And this is the John Innes Centre. It's in the city called Norwich on the outskirts, which is in the eastern part of the country, as Nick said, in an area called East Anglia, somewhat more rural than Leeds. But we have some nice playing fields nearby and things like, and we actually the cricket pitches there as well. So it's a pleasant place to work in a pleasant part of the country. But Leeds also has its attractions, as I know. So the first question is, why on earth, with all these other expression systems, such as E. coli, yeast, insect cells we've heard about, and of course also mammalian cells, would one even consider thinking of plants? I mean, it seems a rather odd kind of expression system. Well, one of the obvious attractions is you can get very large biomass. I mean, plants are used in agriculture, obviously, and that was an attraction initially. They actually require minimal inputs. Obviously, the carbon source is carbon dioxide in the air, so you don't have to add any carbon to any medium. The kind of medium you produce them in is not a complex type of broth. It's can be soil and or an artificial equivalent. And also, actually, if you're talking about a legume, which you can use for certain purposes, even the nitrogen comes from the air. So, you know, theoretically, the input should be low. And of course, the main energy source is sunlight, rather than any other carbon source. I say they keep themselves sterile. What I mean by that is one of the big issues of using a bioreactor is sterility. And bioreactors, complex things, always tend to get contaminated. Whereas plants, actually, there are no bacteria within a plant which is growing in the field, generally. Certainly not any ones which will infect humans within the plant itself. Any product produced in a plant will not be contaminated with an animal pathogen. So that's one of the problems, say, with using mammalian cells for expression. Every so often the cultures tend to crash due to infection with, say, a mammalian virus. Well, you might get a plant virus in a plant, but that is actually harmless to humans. So there has been an attraction. So the idea in the early days, when this was first mooted, was you could go from a rather complex, and this is a very small scale bioreactor, with the broth for growing whatever it is on a small scale in here, to bioreactors which looked a bit more like these. And these are just a rather sort of made up example of maybe growing stuff in rice in the field. Now, it turns out that actually for vaccine production, you don't actually want to grow your plants in an open field because you can't control the conditions very accurately or control any pathogens which might accidentally get involved in it. So we tend to grow everything within greenhouses. But that was an idea, say, from about the 1990s, you might be able to do that. So how would you actually approach expression of foreign proteins in plants? Now, I'm guessing that most people know of the major way that this is done, and that is to create genetically stable lines of plants, transgenic for a gene of interest. And that's widely used, but mainly to create new phenotypes of plants. So a classic one would be to include a gene for herbicide resistance. And one of the most well-known ones is Roundup. So you make your crop plant resistant to Roundup, whereas the adjacent weeds will be killed by the herbicide. Similarly, for things like Bt toxin, you can get varieties of cotton which are transgenic for the Bt toxin, which then prevent the cotton boll weaver, the insect, actually destroying the cotton. So again, and that would just be restricted to the cotton plants. And so transgenic plants have been used to create new phenotypes and are actually widely used now in agriculture. But they're not really designed to produce massive amounts of the foreign protein, just enough to confer the phenotype. They have been explored as a means of making pharmaceutical proteins, mainly antibodies, though, in the early days. Now, the alternative is transient expression of a gene of interest using, well, in this case, and it's where I started, viral vectors, or actually what is much more common now is direct agro-infiltration. And what actually is this transient expression? Because some of you may be aware of it, but others may not be so aware. So I'll just explain it slightly. It is distinct from classic transgenesis. Now, classic transgenesis, actually you take an agrobacterium or plasmid, or you can use biolistic bombardment, and you actually introduce the gene you want to express into the whole genome of the plant. And that can either be the nuclear genome, or in certain instances, it's the plasmid DNA. But basically, you add to the complements of genes within the plants, and then the inserted sequence acts just like a plant gene and is inherited from lines. It goes through the germ line. So you get lines of transgenic plants, which of course is extremely important if you're going to bulk up the number of plants for agricultural use. So you've actually just added a gene to the normal complement of genes, and it's heritable. In the case of transient expression, the foreign sequence is introduced into developed plants. And the sequence does not modify the plant gene in a heritable manner. You just introduce the sequences into somatic cells. And the sequence is actually expressed just transiently in those cells which have received the genes. So you can't collect seeds from those plants, or you can, but they won't be genetically modified. It'll just be the somatic cells which are actually have been in contact with the agrobacterium, which is carrying the gene of interest. But the great advantage is that expression occurs in a matter of days after you've done the introduction of the gene. Whereas getting transgenic plants is quite a long process. Now, depending on the species you're using, it can vary from a matter of months to a matter of years. And one of the problems with that is that if you're going to go down a transgenic approach, you have to be pretty certain in advance that when you finally get the lines of plants expressing the protein of interest, it is going to do what you want. Now, if you're talking about, say, an emerging viral disease, you don't actually have that time to keep on making this long-winded transformation, regeneration, crossing, back crossing, etc. You want to do something much more quickly. So transient expression is much more suitable. But it's essentially a batch process. So you can't just take the seeds of the plants and then you've got, do it once. You have to do the transformation every time you want more material. And the plant most commonly used for this is a plant called Nicotiana benthamiana, which is actually otherwise known as Australian dwarf tobacco. It's related to normal smoking tobacco, but it's actually a considerably smaller plant, but which grows quickly and is very amenable to expressing foreign genes by virtue of the fact that it has a non-functional plant immune system, which has just arisen naturally. So it's very, very suitable. And most of the studies you're going to hear about, well, certainly all the ones in my talk, but often if you hear other people talking about transient expression, it's most likely done in benthamiana. So what actually happens? So I've tried to explain in a generic way what happens. So a few years ago, we made an animation of the process, and I hope that it's got sound and rather cosmic sounding music. But I'll talk you through the process, and I hope this makes things a bit clearer maybe than I have already. So what you do is, you just get this going. You would just isolate a gene from any source. So this just shows DNA, but if we're talking to want to make vaccines, you'll take this from the viral genome. And so you just take the segment of DNA coding for the genes you want. It's for Agrobacterium, but we use plasma, which will also grow in E. coli. So you simply do a straightforward cloning, basic molecular biology, insert your sequence into the plasmid, which will grow in both E. coli for the initial work. And once you've done those manipulations, you grow Agrobacterium tumefaciens, which is a soil-living bacterium, but which can be cultured on plates, and it's motile. And so here you have a view of what Agrobacterium would look like. It's a rather schematic view, but they can move. And what you do is a simple electroporation with your DNA, give the things an electric shock, and your plasmid will actually get into the Agrobacterium and then will amplify. Not to the same extent as it does in E. coli, but you'll get a number of copies. And this is the remarkable bit. All you have to do is grow the Agrobacterium in culture, take it up in a syringe, and simply syringe the solution under the epidermis of your benthamiana leaves, shown here. And you'll flood the leaves. And that's all you have to do to introduce the Agrobacterium, because the bacterium naturally, this is showing inside a plant leaf, see sunlight coming from above, they will swim towards the surface of the plant cells, actually extend a type 4 secretion system, which will allow the plasmids containing your foreign sequence to get into the cytoplasm of the cell. Now these become coated with proteins, which will take them to the nucleus, which is this object here glowing. And these get taken up, and an integration process occurs whereby the bits of the plasmid, by a rather complex process, are integrated into the genome temporarily and transcribed, which appear to make the nucleus go supernova. And these RNA molecules are then translated in the cytoplasm. And we're talking about a Vlp here, so you're making viral subunits, which are purple, as everyone knows. So the patch of your leaf goes purple, and they then self-assemble just by expressing the individual coat proteins within the leaf to produce your Vlp, which can then be extracted and purified. So that's the basis of transient expression. And it is very straightforward, very cheap, and quick to do. using plants grown in soil is for most of our work. And so what are the advantages and disadvantages of this approach using plants? Well, it's extremely quick and easy to perform. So we've gone from designing a sequence of a gene we want, say the coat protein of a virus, and from the day of sending off the sequence to be synthesized by one of the various companies around in the world to actually getting purified Vlps can take about 4 weeks, the whole process. So you can very quickly make material. And of course, the first 2 weeks of that are doing the cloning. So if you want to batch, it probably only takes maybe another 2 weeks for each subsequent batch. The vectors are extremely similar to those used for stable transformation. They're little adjustments in certain cases, but they're basically the same. So you can actually, if you want to go on to stable transformation, you can more or less use the same constructs. If you identify something you really like, you can say, well, I need a large amount of it. I want to use stable transformation. Well, you can then go forth very easily and do that. Disadvantages are the expression only occurs in the areas directly infiltrated. So where the agrobacterium goes, that area of the flooded area of the leaf, that's where it stays. Now that's an advantage in certain cases. It won't go anywhere else. So actually you can infiltrate different patches on the same leaf if you want to look at different types of constructs to compare them. They won't cross-contaminate. But it does mean it can be difficult to get enough material for large-scale production because it's not going to move throughout the plant. And it's therefore very important to maximize the expression levels in the tissues which actually have received the DNA. Now we developed, and this is going back, well, over 10 years ago, a non-replicating system, this Kaplan-Meier virus, CPMV-HT, which is based on a plant virus, but it's not really much of a plant virus anymore. It really just contains components from it. In fact, basically what we do is make an artificial messenger RNA. So the particular virus has 2 genomic components, and the smaller one can't replicate. It contains the genes for the structural proteins, the movement protein, and the large and small coat proteins, but it has these 5' and 3' UTRs. And this actually makes it a very good messenger. You can just use those sequences and put a foreign sequence between them, and in the first case, like virtually everyone else, we use a marker gene, in our case GFP, and all you have to do is then take those constructs, so in a bacterial plasmid, so you're surrounding your gene you want to express with the non-coding regions from Kaplan-Meier virus RNA2, and you just do this infiltration into the Bentamiana leaves, and lo and behold, the bacteria will actually transfer the DNA into the cells and then into the nucleus, and you get expression. Here's the GFP, fluorescence under UV light. But you can actually increase the levels of expression, or basically control them, by altering the sequence of the UTRs. And here's a mutation in the 5' UTR, and suddenly you've got 15 times as much expression. Now, one might think, well, if you can get the higher level, why bother with the lower level? What's the point of that? Well, actually, there is a point, because some proteins can be toxic in plants, and in some cases for complexes, you want to vary the amounts. And I'll give examples of that. So that's very straightforward. A few years ago, 2 PhD students, Ava Tooneman and Frank Sainsbury, in my lab, set to and made some very usable plasmids, the PEAK vector series, and that stands for P, easy and quick, in case anyone's wondering, and because it's designed to make cloning easier. And these were conceived in 2008, 12 years ago now, to allow simple one-step cloning between the 2 UTRs, the 5' and the 3'. You obviously need a promoter for expression and a terminator, and we also put on here a suppressor of gene silencing, and this is, for those of you who know your agrobacterium vectors, you need a right and left border to get the integration events. And you can put modules of these things, line them up on the same tDNA for co-expression of proteins on the multiple vectors, from the same vector. But also, actually, you can co-infiltrate more than one vector, more than one thing at a time to get co-expression. And we actually have kits of these, so if anyone's interested, we actually distribute these kits for expression in plants. And what I find rather remarkable, these original peak vectors, which are now over 10 years old, are still very popular. I mean, we have some upgrades on them, but you know, vectors don't normally last 10 years, but these seem to have, because they're straightforward to use. And as I say, they are available if anyone's interested. And these were published back in 2009. So if you want to scale something, I mentioned small scale stuff on individual plants and even spots on leaves. And there you go. And that's myself in a slightly mocked up picture doing an infiltration, even though, and that's just in a greenhouse. And you grow the plants in soil. These are in Benthamiana plants. And that's on a small scale. I mean, you're never going to be able to scale that up, even with an army of PhD students and postdocs doing it for you. But you can change the process round and do what's known as vacuum infiltration, where you take a whole plant and dump it under the surface of a soup of agrobacterium. And this is just shown in a homemade device here, which will do one plant at a time. And basically what you do is you put the plant below the surface, reduce the air pressure above with a simple vacuum pump, and the air in the intercellular spaces comes out because of the reduced pressure. You can see bubbles forming. You wait a few minutes. When you reduce the pressure, take the vacuum off, that air is replaced by the suspension of agrobacterium. So you do the whole plant simultaneously. And they look a bit bedraggled at first, but they recover within a few minutes, literally. It doesn't in any way really harm the plant at all. But the agrobacterium is throughout the plant now. And that's an industrializable process. And this is a picture taken in a facility in Quebec City by a company called Medicargo, who have an automated system for doing hundreds or thousands of plants at a time. So this is a scalable process. And you can go with exactly the same vector from this individual leaf being infiltrated to kilograms being produced at once. So it's a very scalable process. And that gives you an idea of the yields. So that's a method. Now, actually, I should say you can make lots of different things in using this system. You don't have to make VLPs. You can make enzymes, which can do biotransformations within the plants. I mentioned antibodies, very useful for making those. So it's a widely used system for lots of different applications. But we're concentrating on virus-like particles. And I'm just going to go through this briefly because Nick has already talked about it and we discussed it. VLPs, why do you want to make them? Well, they're non-infectious alternatives to virus particles. Their size is just right to interact with the immune system. They have a repetitive structure, multiple copies of one or a few coat proteins. Again, very good for recognition by the immune system, toll-like receptor ligands. Therefore, ideal as a vaccine candidates. And here just gives you an idea from a review by Bachman and Jennings. They're just a beautiful size. They very accurately mimic pathogenic agents and will react as if they are viruses, whereas soluble proteins tend to be too small to interact efficiently. And VLPs can, in many cases, can simply be made by expressing the subunits. Now, in some of the virus, they'll just self-assemble. Now, in some cases, it's a little bit more complicated because you need a processing step, and Nick talked about Poliovirus, where that's the case. And so I'm going to just give some examples of the kind of things we've made in plants. So these are the kind of things we've made in plants using this system. And for instance, we talked about human papillomavirus. Well, that's obviously well established in a commercial sense. So we've made some bovine papillomavirus particles simply by expressing the single coat protein. We've made particles, VLPs, of another plant virus, and that's for use in nanotechnology. We've made particles of a fish virus, particles of the hepatitis B core particle, which Nick has talked about, and these all have something in common. They're made up of only a single type of subunit. You only have to express a single type of protein, and there's no further processing. You can get more complicated. Blue tongue virus, which is a double-stranded RNA virus, very important in agriculture, infects sheep and to a lesser extent cattle. Papillomavirus virus itself, which is again a nanotechnology particle, and then polio. Now, you might be worried this talk's going to go on forever with all these different things, but I'm just going to give you examples of 3 different ones. And I was very pleased, actually. I was wondering which to choose of the simple ones for any one type of coat protein. And I know that Nick talked about hepatitis B core particles, and so I won't talk about those again. But you can make those in plants as well as E. coli and yeast. And it works very well. And we've worked together with Leeds on that. So the one I've chosen for that is actually, and this relates to agricultural issues, nervous necrosis virus, which is a disease of aquaculture, so it's a disease of fish. And that's very important for feeding people in many parts of the world. I'm going to talk about blue tongue virus, another veterinary vaccine, but I'm choosing that mainly because it's not just one type of coat protein. It's 4 coat proteins need to be expressed in the right ratio to make a particle. And for these non-envelope viruses, I'm also going to talk, and I'm going to keep it brief, I won't give the background because Nick has already given it, poliovirus, but just our experience in plants. So just about nervous necrosis virus, just a little bit of virology. It's a non-envelope virion, 180 subunits of a single type of coat protein, which does actually undergo a catalytic cleavage to release a peptide from the coat protein, but that's not not a very significant maturation step. Although it does naturally occur. So all you have to do, here's the original NNV, which is a member of the nodovirus group of viruses. You simply take the gene for the coat protein, put it into a plasmid, and here we are. Here's the name of the plasmid, our peak plasmid, NNV coat protein, and we've inserted it between the 5' and 3' UTRs, and here's the actual sequence just in here. So we've made that gene. Initially, you do the cloning in E. coli. That's fine. You then amplify the plasmid in coli, and you transform Agrobacterium tumefaciens. So this would all take, from the ordering, and actually, of course, what you do is nowadays you make a synthetic gene. This would all take maybe 2 weeks. You then infiltrate on the small scale initially, and then isolate the particles. And you can isolate the particles. These are very stable particles. It's quite straightforward. You just grind up the plants and use centrifugation. And very straightforward. And you can purify them on gradients if you wish. And you can show that the particles in a Western blot, here they are. This is the protein being nicely expressed, and it reacts with the antibodies. And so you can see that you're making antigenic material, and you can look at the structure of the particles, and this is some nice cryo-EM work done by the Asprey Centre in Leeds to show that our particles look like authentic nodovirus particles, this kind of knobbly appearance, and that's all very nice, but of course this isn't a beauty pageant, just showing how beautiful our Vlps are. They actually have to do something. So, but what about their immunogenicity? And this is something I alluded to. It's very important in all these kind of studies to have collaborators who can do the other bits of the study, because I don't have any access or indeed much knowledge of fish immunology, but we know people who do. And so I'm very grateful to an organisation called CFAS at a place called Weymouth, which is on the south coast of England, who actually study fish diseases. And so they were able to take these fish, which are sea bass, and they have tanks in seawater, and they can husband the fish, produce the fish, and can actually inject them, and this is either intraperinatally or intramuscularly with the VLPs from nervous necrosis virus, and then challenge them with actually live virus to see if it's protected. And if you do this as the control group, you can actually see the poor fish develop symptoms, this downward swimming and this darkening, because this virus, as it implies, attacks the nervous system. And to cut a long story short, when you do these studies, And this is a Kaplan-Meier curve. I don't know how familiar people are with these. I'm not particularly, but they're supplied by our colleagues. These show that you can either do, if you don't vaccinate the fish first, or just to vaccinate them with PBS, fish sadly die quite quickly. If you do vaccinate either intraperitoneally or intramuscularly with the plant-produced vaccine, the survival is much better. So those are initial studies saying that this plant-produced VLP candidate vaccine has the potential to protect fish against disease. Now, this is a very simple type of VLP to make, a single type of coat protein, but it does seem to work. Now, blue tongue virus, on the other hand, is much more complex. 4 main structural proteins form a virus-like particle, and they're arranged in 3 concentric spheres, so a rather elaborate and rather beautiful structure. In the very center, you get a thing called VP3, it's a big protein, 103 kilodalton, and these form trimers in a subcore. On top of those, you get VP7, which assemble onto the subcore, and together these make core-like particles called CLPs, and the structure of these has been known since the 1990s, and a rather beautiful, very stable, nice spiked structure. But the problem is they're not very immunogenic. And so you need to put the 2 outer proteins on. These give triskelion structures on the surface to make a particle like this. This is, there's now a high resolution structure available, but this is the overall architecture. And these are very immunogenic, but they're pH sensitive and they tend to fall apart fairly readily. And there's a real need for vaccines against blue tongue virus because it's a major disease, certainly in sub-Saharan Africa, but also in the Mediterranean basin. And the problem with veterinary vaccines, they have to be really cheap because individual fish in the first case or sheep actually are not that expensive. So you can't have a vaccine which costs more than the animal you're trying to protect. Otherwise, people will just take their chances. And so plants did offer a cheap solution to this. Anyway, so how do you go and make these blue tongue virus particles? Well, you want to express 4 proteins. You can put them all on one construct, which in a way for expression is the most efficient, but it's a bit cumbersome, takes a while to do. Or you can put them on, each one on a separate construct, but then it's quite difficult to control the relative amounts you express. But we found the most efficient and flexible approach was to produce a construct with the 2 inner proteins to make core-like particles and a construct with the 2 outer ones. And there's also a theoretical reason for this, is that there are, well, at the last count, 29 serotypes of BTV, and that's controlled by the outer proteins. And you can make a constant scaffold of the core-like particles with VP7 and VP3 and simply vary the outer ones. And so you don't have to go all the way back to the beginning. And if you do that and do the infiltration of plants with either 1 protein, just the core proteins, all 4, it looks pretty nice. We do it with just the inner one, VP3, you get nice thin-walled subcores. You put the second one, get these lovely core-like particles. You express all 4, and you get a mess. You get some VLPs, look nice, but you get also subcores and core-like particles, and it's not very satisfactory. And the reason for this is you need to control the levels of expression because not all the proteins are present in the same amount. And if you overexpress the central proteins, you get a lot of core-like particles because the equilibrium is very strongly that way. If you only have a few of the outer proteins, you actually find the equilibrium's back towards core-like particles and soluble proteins. So you want to reduce the levels of these inner proteins. And that's where being able to control the level of expression by controlling the sequence of the UTRs really comes into its own. And so we wanted to lower the level of, and we chose VP3, the innermost protein first, remove what's known as the HT mutation to result in a 15-fold reduction. And that's why I said sometimes reducing levels of expression can be beneficial. You really want to do that to get a proper properly formed particle. You don't want everything at the maximum. And we did this by reversing the mutation. And what happens is you can then test the particles, and what you find is you get beautiful virus-like particles in plants, which look very much like authentic BTV particles, and also ones produced in vacuolar virus. And you can carry out sheep immunization experiments. And the details basically here is that if you administer the VLPs and then challenge, you get no disease in this clinical reaction index. So this is actually a vaccine, a potential plant-made vaccine, which actually protects the desired animals, sheep, against blue tongue disease. So it's a question of being able to scale this up for deployment to protect the animals against this disease. So that's expressing 4 proteins simultaneously and controlling the levels to get the appropriate type of particles. Now, I'm going to go briefly through poliovirus VLPs, and Nick has talked about the basics, so I'll just talk about our plant experience. As Nick said, there's a need for a new source of vaccines from the World Health Organization, but as she mentioned, in the absence of RNAs, VLPs are relatively unstable, and they undergo this antigen shift from the D to the C form, and the C form is the one which doesn't raise protective antibodies. So you need to preserve stuff in the D form. So can you make stabilized particles in plants? And to do this, this is the genome that Nick showed, but so I won't go into what exactly it does. You'll need 2 components. You need the coat protein precursor, and you put that in one plasmid, and the protease, 3CD, needed for the cleavage of the mature coat proteins, and you put that in another plasmid. And what happens if you use these and then co-integrate them into plants in the normal matter? You kill the leaves, which isn't what you want, because that's where you rediscover the fact that 3CD, the protease, is actually highly toxic. And so that's not just in plants, that's in lots of systems. So you're not going to get much out of those kind of leaves. And this again is why it's important to control the levels. Because 3CD is an enzyme, you don't need as much of it as you do the structural proteins. So you can reduce its level so that it's no longer toxic, but there's still enough of it to the cleavage of the precursors efficiently. And if you do that, lo and behold, you can get nice particulate looking matter from, and this would be type 1 poliovirus. But this is wild type poliovirus. And as I mentioned, in the absence of RNA, this tends to undergo this antigenic shift to a rather immunologically useless form, the C form. And so we take these nice particles and are very proud of them, and they look okay, and you can purify them. And this is a gradient, density gradient, and you can just take a needle from the side. You can see the layer here where the particles sediment. You can purify them, and you can check that they're, you know, which band the material's in, and look for VP1 of poliovirus. And was this, you know, looked all good, but is this a great success? Well, no. If you do some experiments with our colleagues at NIBSC, you find that everything we've made is in the C antigenic form in this ELISA reaction, and which is the red line, and that's the one you don't want. Compare this with the Salk inactivated poliovirus, where most of the material is in the wanted D form. Now, this is something we sort of expected, because as people have found, that without stabilizing the particles in some way, they do all change to this antigenically irrelevant form. We're very lucky with our collaborators who used forced evolution to select thermostable mutants, and Nick alluded to this. Basically, they simply passaged infectious poliovirus. So not something we could do at the John Innes Centre. It needs a place with category 3 containment, or at least category 2, and licensed to handle live poliovirus. They passaged the virus at increasing temperatures, and then selected the variants which survived and did deep sequencing on them to identify the stabilizing mutations and proved that those viruses were indeed stable. And then all they had to do was provide a sequence file to us. So we never see poliovirus. They simply sent the sequence of the mutant forms. We get those mutant coat proteins synthesized commercially and express them in our plasmid. Replace the wild type P1 region with the mutant form. And of course, we can use the same 3CD protease as the co-inoculum for all of these, because they'll all be cleaved by the same protease. So we can screen a lot of these mutations on plants and see which ones, whether they do enhance stability. And we've done this for all three serotypes. I'm only going to talk about serotype 3, which is the one that Nick didn't talk about. We've had success also with PV1, but we've had problems getting enough material with type 2. And that's actually common in quite a few different expression systems. Anyway, I'll talk about the success first. So here's the material we produced in plants for type 3 polio. First thing we had a look at, again with colleagues in Oxford, the same colleagues that Nick talked about. And we can see we get these nice poliovirus structures, and they appear to be empty, no genome in them. You can do some immunization of mice transgenic for a human poliovirus receptor, and these are the results here. And you can protect the mice against challenge, and you can compare it with IPV. And so you do get protection of mice from polio using VLPs produced in plants. So plants are a legitimate system, but also you'll get the same of the material produced in yeast, and I'm sure with other systems as well. So you can express VLPs where you require a maturation cleavage as well. That will also work in plants, albeit that this normally would only occur in animal cells. Plants will support it. Okay. So then the next question comes, all the viruses I've talked about, although they're of increasing complexity, they're all non-envelope viruses. They don't have a host-derived membrane around them. And some of the most nasty viruses, or many of the very nasty ones we deal with, including the current COVID-19 virus, are actually enveloped. So can you use plants to produce VLPs for envelope viruses? And that's quite a challenge. And so we started a project back in 2016 to look at envelope viruses. Now, of course, we weren't working with COVID-19 then because it hadn't been described. But what we really looked at, first of all, was dengue fever. Now, producing envelope viruses is more complex than non-envelope viruses. Obviously, they contain host components, the membranes. The envelope proteins, the ones that occur in the host-derived membranes, are usually glycosylated, which is not true generally of non-envelope viruses. Maturation also uses host as well as virus-encoded proteases, so it's a more complex cascade, usually. And the correct assembly of the final particles involves interactions between different cellular compartments in a coordinated fashion. So you'll get the internal capsid protein made in the cytoplasm, which then has to meet the host-derived membranes containing the glycosylated envelope proteins at the cell surface. So this is dengue virus, and it's a flavivirus, and has one long polyprotein, and it undergoes cleavages via a signal peptidase, a Golgi protease, that's a host one obviously, and also the NS3, the non-structural protein 3 protease encoded by the genome. And what you need to produce a nice particle, and what you want on the surface, which will have mostly immunologically active sequences, is the envelope protein. This thing's called E here, which occurs on the outer surface. The capsid protein also normally interacts with the genome on the inner surface as well. And that can play a role, but it's mainly the envelope protein. And so to try and make these proteins in plants, we tried all sorts of different things. This is, to try and make this work. And this just shows, actually, one of the benefits of being able to do things very quickly in Benthamyana plants. Lots of different formats. You can try just expressing a precursor of the envelope protein. You can actually then co-express it with the non-structural proteins to get cleavage, to encourage cleavage, so co-expression here. Or you can use mutant versions, with, say, in this case, without the capsid protein, or introduce point mutations. And you can test all these on a small scale and look at which format seems to be best for making VLPs. And if you do this eventually, and after much blood, sweat and tears, to quote Winston Churchill, you can eventually get particles produced, and you can isolate them, and purification is a challenge. And you get the best results if you express precursor plus the non-structural proteins. And these are plant-produced particles made in plants. And you can actually do various other tricks, mutations. There's a particular phenylalanine to alanine, which will increase the amount of proteins you use, VLPs you get, which is useful. And you can actually show that these particles will raise antibodies in mice. Now, it's work in progress, as indicated by the sign, in that we haven't yet had time to do any experiments on protective immunity. But that's the next step, where we can certainly make the proteins of an envelope virus, which is encouraging. So what about making VLPs of SARS-CoV-2? Obviously the causative agent of COVID-19, which is very much in the news all around the world at the moment. Well, we know we can make envelope virus particles, so what would you need to make a VLP of SARS? Well, the genomes of a lot of coronaviruses are known for many years from different animals, and this is, well, it's slightly old. It wasn't called SARS-CoV-2 at the time, but the genome is fairly typical, and this is the genetic structure. It's known, the sequence of SARS-CoV-2 is known. So, and from all the work, mainly on the veterinary coronaviruses, you know what's really required to make a VLP, because you know the structure of the particles, and on the surface is the membrane here. Inside is the nuclear capsid, nuclear protein with the RNA genome, and here are the spikes sticking out on the surface, which are trimers. Now, those are the things which give coronaviruses their characteristic appearance. Obviously, corona means crown, so that's where they get their name. But you also get these other structural proteins, the membrane and the envelope protein, M and E. And so a fair amount was known of what would be the requirements for assembly. So to make them, here's some VLPs for coronavirus. There's the spike proteins. What are you going to need? You're going to need the S, M, and E proteins, though S, the spike protein alone, may be enough. And the spike protein contains most of the epitopes, most of the antigenic sites, as well as those sequences required for receptor binding. And in fact, those are all in the globular domain at the N-terminus of the protein. However, if you just make the globular domain, it's likely to be a soluble protein rather than the VLP. And as I explained, VLPs are generally preferred. Maybe the nuclear protein might be required, as there's evidence that it promotes VLP assembly. You'll need a source of membranes, obviously. It's an envelope protein. Glycosylation apparatus to get glycosylation of the spike protein, in this case, is glycosylated. Now, fortunately, each of the S, M, and E proteins are produced from separate subgenomic RNAs in the virus, making designs of expression cassettes easier. Now, what I mean by that is you've got separate genes. You don't require some kind of proteolytic processing cascade, which was one of the issues with dengue virus. So in a way, just taking the reading frames is fairly straightforward. And so, of course, what you want to do, the first thing you do is, this is a mammalian virus, you tend to always codon optimize the constructs for expression in plants. And one of the other reasons, and then express them. Now, one of the reasons you might want VLPs as well is that some of the major antigenic sites are structural rather than linear. And that's important. And actually, there is evidence from the veterinary coronaviruses that some of them involve interactions between more than one of the structural proteins to create the epitope. So where are we now? And this is work in plants. Well, basically, we took the various genes and expressed them. And this is very provisional, very much work in progress from a postdoc in my lab called Jae-won Young from South Korea. And basically showing that you can get, using Western blots, you can get expression of the spike protein in plants. And also in this case, you can show you get the envelope protein. This is fairly messy because it really is ongoing work. And we've got very, very provisional findings that you get things which look like coronavirus VLPs that you can isolate from plants. But this is all very provisional, so no animal experiments at the moment. So what I'm hoping I've shown you by this is that plant-based expression can be used for producing a variety of different VLPs, both for use in animals in the veterinary context, potentially in humans. You're not limited to simple VLPs containing a single type of co-protein. You can express multiple different proteins, like in the case of Gluten Virus. You can get processing in the case of polio, and you can make enveloped VLPs of enveloped viruses. And I just want to acknowledge the people over the years. This is a picture taken a few years ago on a sunny day in the UK, which does happen occasionally. And these are a lot of the people involved. I'll simply point out Joanna Marzian, who was actually a PhD student, is now working for a vaccine company who's worked on both NNV, the fish VLPs, and also polio. Daniel Pondorf, who also worked on, who's not actually in this picture, oh no, he's there. He's also gone to work for a company. He worked on dengue particularly, and since this slide was made, we had J1 has joined the lab. Also to thank all the cryo-EM collaborators in the Asprey Centre in Leeds, but also particularly I want to thank, of course, the Polio Consortium and colleagues at Leeds, and obviously you've heard from Nick Stonehouse, but also the people at NIBS, National Institute of Biological Standards and Control, who did a lot of the immunology and actually identified the stabilizing mutations. And also, I want to give a call out to the people at the University of Oxford for the cryo-EM analysis. And down here for the NNV, of course, Richard Paley at CFAS, looking after the fish. And I simply want to end up by saying you don't get far without a source of funding. And so we get funding, obviously a lot of the infrastructure is provided by the John Innes Centre and the John Innes Foundation, which is a charitable foundation. But we're essentially publicly funded, but we do have the endowment. We get most of the underpinning money is from the BBSRC, the Biotechnology and Biological Sciences Research Council, but also particularly for polio, we had support from the World Health Organization. And I'd just like to thank you all for listening.
Thank you, Dr. Josh Lomonossoff. That was a very compelling presentation. We are lucky to have you with us. And
well, thank you very much.
We learned a lot from you. Thank you once again. And with that, I would like to request our honorable guest,
Dr. Mohammad Morsal Bella, a former visiting researcher at the University of Edinburgh.
Now a professor of biotechnology and genetic engineering at Columbia University in Bangladesh, to talk a little bit about the career opportunities for our students in virology. So if they choose to do masters or perhaps PhD in virology, what career opportunities they do have and what career opportunities they will have. So Dr. Morsal Bella.
Thank you very much. Thank you, George. Actually, that was an awesome presentation, I should say. Thanks, George. This was extensive and well-coordinated one.
Well, thank you very much. Thank you for your kind comments.
And I have learned so many things, and you have covered so many things so beautifully with this presentation. I have got one question, then I will go to Science Sharker's question. I want to ask, if we want to express viral glycoproteins in plant-based systems, then how can we conduct post-translational modifications or any other processing that that is done in mammalian systems? So can you just tell us how can we do that?
Yeah, well, actually processing, certainly proteolytic processing for mammalian viruses seems to occur quite naturally in plants. Well, certainly if it's a virus encoded which might not be surprising. But also, plants do contain things like furin, or if you haven't got enough of them, you can simply co-express those enzymes by co-infiltration. One of the beauties of it, it's very easy to co-express different proteins, and you can try things quickly. I always say one of the great things of transient expression is you get your failures quickly. And what I mean by that is don't be afraid to try things. You think, well, this might work, but I'm not sure. Well, try it, because it's not going to take you a very long time to assess. If you're trying to make a transgenic plant, you'd be very wary of wasting a year trying to get the lines of plants. Whereas this, you know, a couple of weeks, and you'll know pretty quickly, was that worth pursuing or is that a dead end? And so we've tried all sorts of co-expressions of chaperones and and furins and things. You can put other enzymes in. And the other, of course, major thing about post-translational modification is the glycosylation. Now, plants will glycosylate at the same site. So if you have the N-glycosylation particularly, and that's fine, but they do tend to put slightly different sugars in. So there's no sialic acid in plants. But they also put xylose and fucose in, which may not be optimal. But you can actually just co-express the enzymes for correct glycosylation at the same time. So you can actually add things for adding sialic acid. And people can have co-infiltrated up to 10 constructs in the same patch of leaf simultaneously. They don't tend to interfere with each other, which is quite remarkable. So people have reconstructed whole biosynthetic pathways that way. So you can actually just put other enzymes in if you're having problems with correct modifications. It's not you have to know the enzymes which are responsible, of course. The other thing is that people are doing a lot of genome editing of Benthamiana. So if you have an unwanted reaction, such as the incorporation of fucose, which is a plant-specific sugar, there are lines of plants now available which have that transferase knocked out, and you can do your infiltrations on those edited, genetically edited plants. So you're not stuck with wild-type Benthamiana. So I'm actually involved in an EU project called Mucosiana, which is designed to make, it's a sort of synthetic biology thing, to make different lines of Benthamiana plants which are suitable for the propagation of different, in our case, Vlps.
Okay, thank you very much. Yeah, the thing is that you have explained that very nicely to all of us. In regard to Simon Shorter's question, what I understood from your presentation, that if we want to adopt transient expression system, so in that case, that can be adopted in Bangladesh because it doesn't require any sophisticated laboratory, and it can be done very quickly, and it is also very cheap system. So I think if some of the biotech startups, they want to test some of the things they want to produce, they can easily adopt. And I think also the biopharmaceutical industries in Bangladesh, they can actually come forward and start this type of work. So that I think it will create a venue for our graduates so that they can be employed in that sector. And I think it's very promising sector.
Yeah, and there is another EU project is actually specifically aimed at helping countries adopt the technology and making kits available. It's one of the aims, really, to make, because it's quite interesting, because in say UK or the US, they're very other well-established tech platforms, yeast and E. coli and insect cells. So people are reluctant, maybe, because they're huge facilities. So it's quite difficult to the plants to get a look in. But if there's people who want to start up new things, it may be a good place to start. It's very easy, as long as you have facilities for just growing bacteria, either E. coli or agrobacterium. The rest of it is just plants and soil and a vacuum pump. It's or even a syringe if you're doing it on a very small scale. Growing plants we just use a greenhouse. It's not a growth chamber. And so on a small scale, it's very straightforward. We even supply plasmids with expressing GFP if you want to check that the infiltration's working. So I've supplied these around the world. Benthamiana seeds are extremely easy to get hold of. They're not grown as a crop anywhere. They're a workhorse. So no, I'd encourage that very much so.
Okay, we look forward to, I think Professor Sohail can start some sort of this type of work, and then our students will be encouraged to go for some sort of a startup. And yeah, we look forward to this type of
Well, I'm very happy to help. Very happy to help.
Yeah, thank you very much.
Thank you, Dr. Morsel and Bella. So I'd like to ask Dr. George, is there any downside of this process, of this transient expression system?
Of course, I would say no, it's perfect. But I mean, one of the issues is is scaling up, because it's different than. It's not that it's incredibly difficult. It's just that the technology is different than, say, pharmaceutical companies are used to. You do, when you're purifying the material, you have to get rid of a lot of cellulose, which you don't, debris, which requires slightly different techniques than, say, you would use with yeast or insect cells, certainly mammalian cells. So you need, the grinding up, the breaking up of cells. Otherwise, the purification is, we tend to use centrifugation because that's the kind of small scale stuff. You don't have to. You can use chromatography, and actually we have a collaboration with a small startup, so based on the technology I developed called Vlp Expression Systems, and they tend to prefer chromatography to do it, and some kind of dialysis and the like, because that's more scalable. So all sorts of ways you can purify particles, and we tend to do things, I suppose inevitably, which we're used to. So having worked on viruses all my life, we'll have an ultracentrifuge, but that's not always practical. And ultracentrifuges are not very scalable. Whereas centrifuge, sort of columns, ion exchange columns are, and so you can do it that way. And there's a lot of work has been done on using those kind of methods for purification. The other thing you do have to be careful of, which you'd also definitely have to be careful of with E. coli material, and indeed others, is that, of course, we're infiltrating with agrobacterium into the leaves, and that's a gram-negative bacterium. So it does produce endotoxin, as does E. coli, and endotoxin is a problem. So you do have to test for endotoxin levels, but I think you'd always have to do that anyway, however you produce stuff, because, of course, endotoxin doesn't necessarily have to be part of the production system, but it's very easy to get endotoxin contamination in from water or wherever, if there's any source of bacteria. So you have to do that kind of monitoring. Obviously, you can do such kind of things as quality assurance. There are various ways of measuring the size of particles and their effect. But generally, it's been, I found it, again, it's me, very straightforward. I think, I've sent the vectors around the world, and people use for all sorts of things. A lot of it's to do with using enzymes rather than Vlps, but they are widely used. And we also have, I suppose, the peak 2.0 type things as well, which, do essentially the same thing, but slight improvements with cloning and the like.
So there was another question. What types of plants can we use? Can we use all sorts of plants for transient expression?
You can. People have thought, wouldn't it be great to use edible plants? Lettuce we've tried, all sorts of things, and they work. But nothing yet has worked quite as well as Benthamiana. It just, because the thing about it is, it's a plant which was collected, actually, just a bit of history, on the third voyage of HMS Beagle. Now, that's a ship that took Darwin to the Galapagos, and it was a naval vessel. But on its third voyage, it went to Australia, and it collected this rather, it doesn't look like much, Benthamiana, but it has an odd fact, because of its lifestyle, shall we call it, in the desert. It's not subject to viral infection, naturally. And so its immunity to virus infection and to expressing foreign proteins, in other words, its gene silencing mechanism, is more or less non-functional. Therefore, it doesn't resist the expression.
Okay, there was another question. That is there any vaccines that uses Vlp for coronavirus in development, and that is promising as well?
Oh, absolutely. Absolutely, there are. I mean, I think Nick said something like 30. Yeah, so VLPs, I mean, you hear things, I mean, I know the VLP-based ones in France, I think in Russia as well, although I'm not totally clear what they are. I think in China, and that's the more traditional approach to go for VLPs. Now also, there's also work reaching phase 1 trials of coronavirus VLPs produced in tobacco. And the company, so tobacco companies who are trying to turn over a new leaf, as we say, are trying to sort of, it's good PR, and they're not just making smoking materials.
Yeah, they're trying to compensate the
Yes,
things.
exactly. To do that. So Philip Morris or British American Tobacco, they're looking at them, and they've certainly made VLPs. So no, they're very much into that kind of thing. So they are being looked at. Now, I think it's good that a number of different approaches are being made, because you can never tell which is going to be, which of any, I have to say, is going to be the most effective. Because, there's been many potential vaccines, which haven't made it. And of course, there are also the very new technologies, such as the University of Oxford here, this adenovirus-based system, and there's an RNA vaccine system developed by Imperial College. Those are the ones in the UK that I know about. But I mean, there is no RNA-based vaccine ever licensed. So I think what's going to happen with this, looking at the upside, if there is, you can consider an upside of the current pandemic, is that it's stimulated a lot of work in methods of vaccine production, and lots of things are being tried. It's also been quite a showcase for how quickly you can get from identifying a pathogen to expressing material, to get immunogenic material. Of course, the one thing it's difficult to get the time scale down on is the clinical trials, because humans and indeed other animals take a certain time to develop an immune response, and obviously you have to do all the safety trials as well.
So there is another question. So within the two approaches, edible plant vaccines and plant-derived virus-like particles, which one is more effective as a vaccine to human, and why?
So, I mean, edible vaccines, I mean, there's been a lot of hope that you could actually just eat plants. That would likely only work if the virus was an enteric virus. I could infect via the intestinal mucosa. So, I mean, whether you could ever use that route to protect against a respiratory virus, such as SARS-CoV-2, is a question. Yeah, so it's an interesting, at the moment, most of my work, yes, all of it has been done with the sort of parenteral administration. But I mean, there's a real hope. I mean, it'd be fantastic, for instance, with the fish vaccine, if you could just make plant pellets, make fish food. Because actually, in practice, injecting fish individually, and I have to say, I've seen film of how it is done on a industrial scale, which is quite amazing. So 10,000 fish an hour. And they don't treat them. They treat them rather rough, I have to say. But it's an amazing machine seeing that in action. On film, I haven't literally seen it. But actually, obviously, you'd much prefer just to be able to put some fish food, some formulation of plant material, which the fish might like to eat, and immunize that way. So, yeah, I think there's definitely an interest in edible vaccines. And one of the problems in the past has been controlling the dose. Would you get enough of it?
Well, thank you, Dr. George. So there was another question, that is, should we immunize the animals?
Oh, yeah, I mean, the animals are definitely immunized. The problem is the cost of doing it, because it does cause a lot of suffering. And I mean, for instance, blue tongue virus, there is a live attenuated vaccine used in South Africa, and the trouble is it's not perfect, and actually also potentiates spread. So it is partially used, but no, vaccines are definitely used in the veterinary field. There's a lot of interest, for instance, in foot and mouth disease virus, which is related to polio. And because, it would be wonderful if you could actually vaccinate cattle against that. There's obviously we did some work on bovine papillomavirus as well. So no, there is a lot of interest. Plus, if you could do it, it's a way of reducing the amount of virus. If you're talking about zoonotic viruses, if you could, and it's difficult to, I mentioned it's difficult to do wild animals, like trying to vaccinate all the bats for SARS-CoV-2 is not really practical. But if it was, sort of livestock, that may well help. And I'll give an example. I think every year we have outbreaks of influenza, and a lot of those are thought to originate in either poultry or pigs. And if you could somehow reduce that and the rate of adaptation to humans, but you have to make, for animals, you have to make it very low cost, otherwise it won't be taken up.
Thank you, Dr. George. Dr. Abdullah, sir, can you hear me?
He is not with us right at this moment. I can see him now.
So Dr. Sir is not there.
We had a lot of questions, but most of the questions have been answered by Dr. George during the lecture.
So we don't have a lot of the questions left now. So if there is a COVID-19 vaccine, Bangladesh will get it or not? So there was a question like this.
Well, I would hope it would be made available worldwide. I'd certainly hope that, and I'm sure that's what the WHO hopes. I mean, there has been sometimes what looks like an unseemly scramble, and that is an issue actually with all vaccines. If there is a shortage of supply, who might get it first? And that is something to be addressed. And of course, but it might not just be one producer. There might be a number of different people, agencies who produce it. So one hopes that it would be used worldwide. And in fact, I mean, even if you were, in a developed country, say UK, USA, or whatever, highly developed country, you're only ever going to, it's not going to be no good just to eliminate the virus in those countries, because it's just going to come back. I mean, it spreads worldwide. It has to be a concerted worldwide campaign. It's silly to think you can just have a nationalistic view of it, a purely nationalistic view. It doesn't make any sense.
So I'll request Dr. Mursal and Bilal, sir, to highlight some of the career options that biotech graduates can have
and do have right now.
We are joined by Dr. Abdullah Malashwal, sir. So I will go with Dr. Abdullah Malashwal, sir. So, sir, do you have any questions for George?
Yes. Sorry, I was disconnected. Hi, George. My question has come from our guest. One is WHO recently that it may happen we never find any suitable vaccine for COVID-19. WHO stated recently. Well, this question
I couldn't quite catch because you're freezing occasionally.
Can I just supplement Professor Sohail? So he was asking that in recent times, WHO, World Health Organization, has made a statement that there have been any reliable treatment options for COVID-19. There could not be any suitable vaccine for COVID-19. What is your opinion regarding this?
Well, I think they were talking about treatments. In other words, trying to say there's all this talk about hydroxychloroquine, and that's been touted, and that's saying that's not really appropriate. I mean, there are things like dexamethasone, which alleviates the symptoms for very sick people, and that's I don't know if you quite call it a treatment. I mean, the treatment for the symptoms, and also remdesivir. Developed with Ebola for inhibiting RNA. So, but I mean, they're not really appropriate for wide-scale use, only for people in severely affected people in hospital. So I think the hope is that a vaccine or multiple vaccines, there may be more than one, is the hope for the future. And we hope that we're in a situation that it should be possible to develop, and there's a lot of background information on coronaviruses, from the veterinary field, that this should actually be possible and to immunise. So I'm hopeful that at least one of the approaches will actually be effective. But I mean, of course, you can never be 100% sure.
So I'd like to add to this. There is a popular opinion that we might never have a working vaccine against COVID-19, just like we don't have a working vaccine against flu, the influenza, so it changes over time. So there is an opinion like that. So what's your say on that?
Oh, now that's a very good point indeed. I mean, obviously there are influenza vaccines, but of course the virus mutates, reassorts to escape immune surveillance. And so it's always a race every year to create a new vaccine incorporating the currently circulating strains. And it's possible we could be in an influenza situation. We're always having to produce new versions. The one thing which gives you a bit more hope, shall we say, is that coronaviruses tend not to vary quite as much as influenza. And one of the reasons for that is that it only has one genetic segment, whereas influenza has 8 in most cases. And so you get reassortments. If you get co-infections, they swap segments. Well, that doesn't happen to the same degree with coronavirus. Also, the replication of coronavirus RNA seems to be more faithful. And what I mean by that is you tend to produce fewer mutants. And of course, it's the mutants which accumulate, which give you the variants. And so it does appear that mutation rate is lower with coronaviruses. Therefore, you may not get new antigenic variants arising quite as quickly. But I think definitely, I mean, it's not just be one vaccine forever and that'll be it. I think one would have to always do disease surveillance and look at the circulating strains. Because yes, certainly, we've already seen it. We had SARS-CoV-1, which caused SARS, and this is definitely related to it. And you can try, give the phylogenetic trees. So new variants can emerge. And so, but I think what you want to do is develop a system for making vaccines which is generic. So we know which bits, say, of the spike protein to put in. We know to put the spike protein in. And so we put it in for one strain, but you could quickly substitute just that bit for the other. And if you know that approach is safe, you don't have to go through all the safety tests every year, which is what happens with influenza. You don't have to start from scratch every year. You know you're using this approach. That generic approach is approved. And so you just have to make the modified version. And that's also automatically approved.
That becomes easier. So there is another question from Tropical Islam to Dr. Professor George. Thank you for your nice presentation. Is there any difference in terms of antigenic nature of the protein produced in plant system and animal cells? So is there any antigenic difference between the protein produced in plant system and animal system?
Well, broadly, they're similar. The main thing which might be slightly different is if you're talking about glycoproteins, because the sugar residues put in can differ. And so if the antigenic site is made up of the glycan section, they could be differences. But there are ways of tackling it by using these edited lines of Pentamiana. Actually, plants have something unexpected about them, which we've only really just discovered. But often when you isolate Vlps from plants, they tend to be naturally somewhat cross-linked because of the peroxidases present in plants. And actually that helps the immunogenicity of the plant-produced material because it stabilizes the particle. So often vaccines actually have some cross-linking introduced deliberately during manufacture to help stabilize it. It turns out that the plant-produced material often has that cross-linking automatically in it due to plant enzymes, which is an unexpected bonus.
Thanks, Doctor. You are muted.
Thank you, Dr. George. So we are joined by Dr. Abdullah also. So, sir, do you have any more questions?
Okay, from participants, no more questions, but I have some supplementary question. George, someone asked that can we use plant, tree, in big plant for this purpose?
So if I heard that, can you use big plants like trees?
Trees, yeah.
Yeah, in theory you could. I think vacuum infiltration might be quite tricky. Trying to take a tree and put it in a vat. But you could imagine the leaves. I don't see why you couldn't use a perennial and just, you can detach the leaves of the plant and vacuum infiltrate them. So you can have a perennial plant and just every so often harvest the leaves and put them in the vacuum infiltration device. I mean, that's the. I think whole trees might be somewhat impractical. Now, people have actually used transgenic trees. Now, that's a different thing where you actually alter the germline and it's heritable. But that's mainly been done for things to do with bioremediation rather than vaccine production or that kind of thing. Because, of course, the regeneration and getting trees to the flowering stage takes quite a long time. And that's rather puts people off the scaling up.
Dr. George, our participants almost.
I've lost that.
I cannot listen to you, sir. I think there is some connection problem from Dr. Lomonossoff's side. So I will go to or another professor, Dr. Mursal and Bella, sir. So if you could enlighten us about the career opportunities that we might have as biotech graduates. We don't have a lot of career options in Bangladesh right now.
Well, I imagine actually, I would say virologists have never been so popular. And remarkably, I'm having to do often more than once a day radio interviews about the virus and coronaviruses and vaccines. So I think if you're interested in the field of virus vaccinology, now is a very good time to go into that field. And you don't have to have had experience immediately. There's so many different variants on aspects of it. There's obviously the production of, say, Vlps, or indeed other approaches, expression studies, purification studies, but also, of course, immunological studies and understanding how the immune system results. So I think, the biotech side, now I obviously sing the praises of plants, and there are biotech companies set up in plants to exploit plants, but of course there are other ones as well. And yet I think more of the more larger pharmaceutical companies. But I would say that would be actually a very good time to be looking into this. I'm sure if you want a simple system, so within Bangladesh, plants would be a good system to try, and the vectors are available. When plants is not particularly difficult, but also I'm sure bacteria or yeast, that Nick said, was slightly more complicated. But no, it's and maybe even contact people in different countries to see. It's I think now if you're interested in that field, people have become far more aware of viral diseases and the need for new vaccines. If you were to talk, say, 20 years ago, people said, "Well, that's all solved." Why do we need any new vaccines? Everything's fine. And obviously, nobody says that now. That sounds ridiculous, and you never know what's going to emerge. I think that's the issue. It's suddenly become, after first of all SARS, then MERS, and Ebola keeps on re-emerging. There's dengue virus is a problem, chikungunya, a whole series of emerging viruses.
Okay, sir. Dr. Abdullah, sir, can you hear me?
I can. Oh, sorry.
Dr. Shral, sir. Dr. Mosul and Bilal, sir.
Okay. Yeah, I can hear you.
Okay, then we'll have to enter the loving us.
Okay. The thing is that I can fill in the gaps by this time before you right. So the thing what happens. So actually, the COVID-19 situation has shown the country that there are lots of areas where biotech graduates can work. So most of the first comes the vaccine industries. In Bangladesh, vaccine industries are in operation, like the insect, and we have seen that another biotech industry that is called Globe Biotech. They have also come into the scene, and they have claimed that they are just producing some sort of vaccine against COVID-19. So I think this is one of the area where the biotech graduates can work. And if they can adopt some platform like plant-based system as a molecular farming technique for the mass production of Vlps, or that could be really feasible option for countries like Bangladesh. And another thing is that the diagnostic industries right at this moment, they are in the full bloom, I should say. So the thing is that our graduates can easily opt for this diagnostic industries, like diagnostic. They can actually do some because they have, I should say, sound knowledge one RT-PCR and all sorts of aspects, and there can be also a scope of rapid detection devices. So a research organization, they can also come forward, like NIV and other related organizations, they should come forward, and they can actually employ biotech graduates, because biotech graduates, they know immunology, biochemistry, molecular biology, everything better than other graduates, I should say. And they have so I think there are many opportunities that biotechnology graduates can opt for. So I'm not sure whether Sohail is back. Professor Sohail is back.
Sir, can you hear me, Dr. Shaw, sir?
I could just add in, I absolutely agree. The diagnostic field is extremely important. And you can make rapid progress on that because it's all in vitro. I mean, you don't have, obviously you have to quality assure that the thing does what it says, but you're not going to inject it into people. So it's nothing like the lead time or the money. So I've actually had a programmer, John Innes, just making controls for PCR-based methods. So no, and that's a very good point, actually, going to the diagnostics field as well, because it's testing and tracking of infections is incredibly important.
Thank you, Dr. Josh, for your compelling reply. Also, Dr. Abdullah, sir, are you with me?
Yes. Oh, yes, I am with you.
Yes, I can hear you. Do you have any more questions?
Okay. Thank you, Dr. Mursaleen, because I think almost all questions and comments already raised. And I'd like to thank you, Dr. George. And we are really looking forward hearing from you. And we, of course, will do some research collaboration with you definitely. As Dr. Mursaleen said, some startup company or biotech startup can come forward for this plant-based vaccine, and we will be the communicator or we will be the collaborator between you and them, so we can start and we can give space for biotech graduates to do more research and others. So that's all I think from my side. It's already 10 p.m. and you are with us more than 3 hours. Really appreciate it. Thank you so much.
A huge thank to you, Dr. Josh. A huge thank to you for being here for more than 3 hours, and we want you by our side. So if you do anything, if you do any startup, if we start any startup, we want you by our side.
Well, thank you very much, and I very much enjoyed it. And I thank you for your stamina in staying up so long to 10 o'clock. I'm sure you have other things you wish to do with the rest of your evening.
No. It's just absolutely stunning. You were stunning. Okay, thank you. Thank you, everybody.
Okay.
That's all.
Thank you, everyone.
And good evening, Dr. Josh. Good evening, Dr. Mursaleen Billah, Dr. Abdullah. Thank you, everybody. It's a pleasure.